Mycobacterium tuberculosis causes tuberculosis. Streptomyces produces streptomycin as well as hundreds of other antibiotics. We derive most of our antibiotics from members of the actinocytes. Another interesting group of firmicutes, the mycoplasmas, lack cell walls, although some have a stiffening material outside the plasma membrane. Some of them are the smallest cellular creatures ever discovered. They are even smaller than chlamydias. The smallest mycoplasmas capable of multiplication have a diameter of about 0.2 micrometers. They are small in another crucial sense as well. They have less than half as much DNA as do most other prokaryotes, but they still can grow autonomously. It has been speculated that the amount of DNA in a mycoplasma may be the minimum amount required to encode the essential properties of a living cell. The domain archaea consists mainly of prokaryotic genera that live in habitats notable for characteristics such as extreme salinity, salt content, low oxygen concentrations, high temperatures, or higher low pH. However, many archaea live in habitats that are not extreme. Perhaps the largest number of archaea live in the ocean depths. On the face of it, the archaea do not seem to belong together as a group. One current classification scheme divides the domain into two principal groups, Euryarchaea and Chronochaeta. In fact, we know relatively little about the phylogeny of archaea, in part because the study of archaea is still in its early stages. We do know that archaea share certain characteristics. The archaea share some unique characteristics. Two characteristics shared by all archaea are the absence of peptidoglycan in their cell walls and the presence of lipids of distinctive composition in their cell membranes, CTO1. The base sequences of their ribosomal RNA support a close evolutionary relationship among them. Their separation from the bacteria and eukarya was clarified when biologists sequenced the first archaeal genome. It consisted of 1,738 genes, more than half of which were unlike any genes ever found in the other two domains. The unusual lipids in the membranes of archaea deserve some description. They are found in all archaea and in no bacteria or eukaryotes. Most bacterial and eukaryotic membrane lipids contain unbranched long-chain fatty acids. Most bacterial and eukaryotic membrane lipids contain unbranched long-chain fatty acids connected to glycerol by ester linkages. In contrast, some archaeal membrane lipids contain long-chain hydrocarbons connected to glycerol by ether linkages. In addition, the long-chain hydrocarbons of the archaea are branched. One class of these lipids, with hydrocarbon chains 40 carbon atoms in length, contains glycerol at both ends of the hydrocarbons, figure 5, left. This lipid monolayer structure, unique to the domain archaea, still fits in a biological membrane because the lipids are twice as long as the typical lipids in the bilayers of other membranes, see figure 5. Lipid monolayers and bilayers are both found among the archaea. In spite of this striking difference in their membrane lipids, all three domains have membranes with similar overall structures, dimensions, and functions. Most Chronochaeta live in hot, acidic places. Most known Chronochaeta are both thermophilic, heat-loving, and acidophilic, acid-loving. Members of the genus Sulfolobus live in hot sulfur springs at temperatures of 70 to 75 degrees C. They die of cold at 55 degrees Celsius, 131 degrees Fahrenheit. Hot sulfur springs are also extremely acidic. Sulfolobus grows best in the range from pH 2 to pH 3, but it rarely tolerates pH values as low as 0.9. One species of the genus Ferroplasma lives at a pH near 0. Some acidophilic hydrodomophiles maintain an internal pH near 7, neutral, in spite of their acidic environment. These and other hydrodomophiles thrive where very few other organisms can even survive. The Euryarchaea live in many surprising places. Some species of Euryarchaea share the property of producing methane, CH4, by reducing carbon dioxide. All of these methanogens are obligate anaerobes, and methane production is a key step in their energy metabolism. Comparison of rRNA nucleotide sequences reveal a close evolutionary relationship among all these methanogens, which were previously assigned to several unrelated bacterial groups. Methanogens release approximately 2 billion tons of methane gas into Earth's atmosphere each year, accounting for 80 to 90% of the methane in the atmosphere, including that associated with mammalian belching. Approximately a third of this methane comes from methanogens living in the guts of grazing herbivores such as cows. Methane is increasing in Earth's atmosphere by about 1% per year and is a major contributor to the greenhouse effect. Most of the increase is probably due to increases in cattle and rice farming and the methanogens associated with both. One methanogen, Methanopyrus, lives on the ocean bottom near blazing hydrothermal vents. Methanopyrus can survive and grow at 110 degrees Celsius. It grows best at 98 degrees Celsius and not at all at temperatures below 84 degrees Celsius. Another group of Euryarchaea, the extreme halophils, salt lovers, lives exclusively in very salty environments. Because they contain pink carotenoids, they can be seen easily under some circumstance. Halophils grow in the Dead Sea and in brines of all types. Picklefish may sometimes show reddish pink spots that are colonies of halophilic archaea. Few other organisms can live in the saltiest of the homes that the extreme halophils occupy. Most would dry to death, losing too much water to the hypertonic environment. Extreme halophils have been found in lakes with pH values as high as 11.5, the most alkaline environment inhabited by living organisms, and almost as alkaline as household ammonia. Some of the extreme halophils have a unique system for trapping light energy and using it to form ATP, without using any form of chlorophyll, when oxygen is in short supply. They use the pigment retinol, also found in the vertebrate eye, combined with a protein to form a light-absorbing molecule called bacteriorhodopsin, and they form ATP by a chemiosmotic mechanism. Another member of the Euryarchaea, Dermoplasma, has no cell wall. It is thermophilic and acidophilic, its metabolism is aerobic, and it lives in cold deposits. It has the smallest genome among the archaea, and perhaps the smallest along with the mycoplasmas of any free living organism, 1,100,000 base pairs. In addition to these archaea that are found in amazing habitats, many Chronochaeta and Euryarchaea live in environments that are not extreme. Protists. Many modern members of the eukarya, trees, mushrooms, and dogs, not to mention ourselves, are familiar to us. We would have no problem recognizing these organisms as members of the kingdoms Plantae, fungi, and Animalia. However, amoebas and a dazzling assortment of other eukaryotes, mostly microscopic organisms, don't fit into these three kingdoms. We call all those eukaryotes that are neither plants, animals, nor fungi protists. The protists are not a clade, they are a polyphyletic group. Some protists are more closely related to the animals than they are to other protists. Some protists are modal, while others are stationary. Some are photosynthetic, while others are heterotrophic. Most are unicellular, while some giant kelps are not only multicellular but also huge, sometimes achieving lengths greater than that of a football field. The protists include some of the most ancient eukaryotic organisms as well as the ancestors of the plant. The protists include some of the most ancient eukaryotic organisms as well as the ancestors of the plants, animals, and fungi. The origin of the eukaryotic cell was one of the pivotal events in evolutionary history. The origin of the eukaryotic cell. The eukaryotic cell differs in many ways from the prokaryotic cell. How did it originate? Given the nature of evolutionary processes, the differences cannot all have arisen simultaneously. We think we can make some reasonable inferences about the most important events, bearing in mind that the global environment underwent an enormous change, from anaerobic to aerobic, during the course of these events. As you read this chapter, keep in mind that the steps we suggest are just that, reasonable inferences. This version of the story is one of a few under current consideration. We present it as a framework for thinking about this challenging problem, not as a set of facts. The modern eukaryotic cell arose in several steps. The essential steps in the origin of the eukaryotic cell include the origin of a flexible cell surface, the origin of a cytoskeleton, the origin of a nuclear envelope, the appearance of digestive vesicles, the endosymbiotic acquisition of certain organelles. What a flexible cell surface allows. Many ancient fossil prokaryotes look like rods, and we presume that they, like most present-day prokaryotic cells, had firm cell walls. The first step toward the eukaryotic condition may have been the loss of the cell wall by an ancestral prokaryotic cell. This may not seem like an obvious first step, but consider the possibilities open to a flexible cell without a wall. First, think of cell size. As a cell grows larger, its surface area to volume ratio decreases. Unless the surface area can be increased, the cell volume will reach an upper limit. If the surface is flexible, it can fold inward and elaborate itself, creating more surface area for gas and nutrient exchange. Figure 1. With a surface flexible enough to allow infolding, the cell can exchange materials with its environment rapidly enough to sustain a larger volume and more rapid metabolism. Further, a flexible surface can pinch off bits of the environment, bringing them into the cell by endocytosis. Figure 2. The chromosome of a bacterial cell is attached to a site on its plasma membrane. If that region of the plasma membrane were to fold into the cell, the first step would be taken toward the evolution of a nucleus, the key feature of the eukaryotic cell. What are some other likely early changes? Changes in cell structure and function. Other early steps in the evolution of the eukaryotic cell are likely to have included three advances: the formation of ribosome internal membranes, some of which surrounded the DNA, see figure 2, the appearance of a cytoskeleton, and the evolution of digestive vesicles. A cytoskeleton made up of actin fibers and microtubules would allow the cell to manage changes in shape, to distribute daughter chromosomes, and to move materials from one part of the now much larger cell to other parts. The origin of the cytoskeleton remains a mystery, heightened by the fact that the genes that encode most of the cytoskeleton are present in neither bacteria nor archaea. An intriguing and controversial suggestion is that a fourth domain of life, now long extinct, originated these genes and transferred them laterally to an ancestor of the early eukaryotes. From an intermediate kind of cell, the next advance was probably to a cell that we could call a phagocyte, a model cell that could prey on other cells by engulfing and digesting them. The first true eukaryote possessed a cytoskeleton and a nuclear envelope. It may have had an associated endoplasmic reticulum and Golgi apparatus, and perhaps one or more flagella of the eukaryotic type. Endosymbiosis and organelles. While the processes already outlined were taking place, the cyanobacteria were very busy, generating oxygen gas as a product of photosynthesis. The increasing O2 levels in the atmosphere had disastrous consequences for most other living things because most organisms at the time, archaea and bacteria, were unable to tolerate the newly aerobic, oxidizing environment. But some prokaryotes managed to cope with these changes, and, fortunately for us, so did some of the ancient phagocytes. According to one highly speculative hypothesis, the key to the survival of early phagocytes was the ingestion and incorporation of a prokaryote that took up residence within the phagocyte and evolved into the peroxisomes of today. See figure 2. These organelles were able to disarm the toxic products of oxygen action, such as hydrogen peroxide. This association may have been the first important endosymbiosis in the evolution of the eukaryotic cell. A crucial endosymbiotic event in the history of the eukaryote was the incorporation of a proteobacterium that evolved into the mitochondrion. Upon completion of this step, the basic modern eukaryotic cell was complete. Some very important eukaryotes were the result of yet another endosymbiotic step, the incorporation of a prokaryote related to today's cyanobacteria, which became the chloroplast. Many uncertainties remain. Several uncertainties cloud our current understanding of the origins of eukaryotic cells. Lateral gene transfer complicates the study of eukaryotic origins, just as it complicates the study of relationships among the prokaryote clades. At the same time, it may not have been extensive enough. At the same time, it may not have been extensive enough to account for the fact that, as genetic studies advance, more and more genes of bacterial origin are being found in eukaryotes. An endosymbiotic origin of mitochondria and chloroplasts accounts for the presence of bacterial genes encoding enzymes for energy metabolism, respiration, and photosynthesis, but it does not explain the presence of many other bacterial genes. The eukaryotic genome clearly is a mixture of genes with two distinct origins. Erison's suggestion is that the eukaryote might have arisen from the mutualistic fusion, not endosymbiosis, of a gram-negative bacterium and an archaean. There are many interesting ideas about eukaryotic origins awaiting additional data and analysis. General biology of the protists. Most protists are aquatic. Some live in marine environments, others in fresh water, and still others in the body fluids of other organisms. The slime molds inhabit damp soil and the moist, decaying bark of rotting trees. Many other protists also live in soil water, some of them contributing to the global nitrogen cycle by preying on soil bacteria and recycling their nitrogen compounds into nitrates. Most protists are unicellular, but some are multicellular, and a few are very large. Protists are strikingly diverse in their structure, but not so diverse in their metabolism as the prokaryotes, which is not surprising, since some of the eukaryotes' most important metabolic pathways were borrowed from bacteria through endosymbiosis. However, protists do display a number of nutritional modes. Some are photosynthetic autotrophs, some are heterotrophs, and some switch with these between the autotrophic and heterotrophic modes of nutrition. Some protists, formerly classified as animals, are sometimes referred to as protozoans, although biologists increasingly regard this term as inappropriate because it lumps together protist groups that are phylogenetically distant from one another. Most protozoans are ingested heterotrophs. Similarly, there are several kinds of photosynthetic protists that some biologists still refer to as algae singular, alga. Although these two terms are useful in some contexts, they do not correspond with natural phylogeny, and we generally avoid them in this book except as parts of descriptive names such as brown algae. Let's next consider some of the other ways in which protists differ from one another. Protists have diverse means of locomotion. Although a few protist groups consist entirely of non-motile organisms, most groups include cells that move, either by amoeboid motion, by ciliary action, or by means of flagella. In amoeboid motion, the cell forms pseudopods, false feet, that are extensions of its constantly changing body mass. Cells such as the amoeba simply extend a pseudopod and then flow into it. Cilia are tiny, hair-like organelles that beat in a coordinated fashion to move the cell forward or backward. A eukaryotic flagella moves like a whip. Some flagella push the cell forward, others pull the cell forward. Cilia and eukaryotic flagella are identical in cross-section, they differ only in length. Vesicles perform a variety of functions. Unicellular organisms tend to be of microscopic size. As we noted above, an important reason that cells are small is that they need enough membrane surface area in relation to their volume to support the exchange of materials required for their existence. Many relatively large unicellular protists minimize this problem by having membrane-enclosed vesicles of various types that increase their effective surface area. Organisms living in freshwater are hypertonic to their environment. Many freshwater protists address this problem by means of specialized vesicles that excrete the excess water they constantly take in by osmosis. Members of several protist groups have such contractile vacuoles. The excess water collects in the contractile vacuole, which then expels the water from the cell. Figure 3. A second important type of vesicle found in many protists is the food vacuole. Protists such as paramecium engulf solid food by endocytosis, forming a food vacuole within which the food is digested. Figure 4. Smaller vesicles containing digested food pinch away from the food vesicle and enter the cytoplasm. These tiny vesicles provide a large surface area across which the products of digestion may be absorbed by the rest of the cell. The cell surfaces of protists are diverse. A few protists, such as some amoebas, are surrounded by only a plasma membrane, but most have stiffer surfaces that maintain the structural integrity of the cell. Many protists have cell walls, which are often complex in structure. Other protists that lack cell walls have a variety of ways of strengthening their surfaces. Some have internal shells, which the organism either produces itself, as foraminiferans do, or makes from bits of sand and thickenings immediately beneath the plasma membrane, as some amoebas do. Many protists contain endosymbionts. Endosymbiosis is very common among the protists, and in some instances both the host and the endosymbiont are protists. Many radiolarians, for example, harbor photosynthetic protists. As a result, these radiolarians appear greenish or golden, depending on the type of endosymbiont they contain. This arrangement is beneficial to the radiolarian, for it can make use of the organic nutrients produced by its photosynthetic guest. The guest, in turn, may make use of metabolites made by the host, or it may simply receive physical protection. In other cases, the guest may be a prisoner, exploited for its photosynthetic products while receiving no benefit itself. We will take a more detailed look at the history of endosymbiosis among photosynthetic protists later in this chapter. Both asexual and sexual reproduction occur among the protists. Although most protists practice both asexual and sexual reproduction, some groups lack sexual reproduction. As we will see, some asexually reproducing protists also engage in genetic recombination that does not directly result in reproduction. Asexual reproductive processes in the protists include binary fission, splitting of the cell, with mitosis followed by cytokinesis, multiple fission, splitting into more than two cells, budding, the outgrowth of a new cell from the surface of an old one, and the formation of sporced cells that are capable of developing into new organisms. Sexual reproduction also takes various forms. In some protists, as in animals, the gametes are the only haploid cells. In some other protists, by contrast, both diploid and haploid cells undergo mitosis, giving rise to alternation of generations, which will be described later in this chapter. The diversity of form, habitat, metabolism, locomotion, reproduction, and life cycles found among the protists reflects the diversity of avenues pursued during the early evolution of eukaryotes. Many of these avenues led to great success, judging from the abundance and diversity of today's protists and other eukaryotes. Protist diversity. The phylogeny of protists is an area of exciting, challenging research. The marvelous diversity of protist body forms and nutritional lifestyle seems reason enough for a fascination with these organisms, but questions about how the multicellular eukaryotic kingdoms originated from the protists stimulate further interest. Fortunately, the tools of molecular biology, such as rRNA sequencing, are making it possible to explore evolutionary relationships among the protists in ever greater detail and with greater confidence. We will discuss several protist clades in this chapter, as well as a few other groups of more uncertain phylogenetic status. Some biologists refer to many of these clades as kingdoms, others refer to them as subkingdoms, and still others refer to them as phyla. This choice of words is not of immediate concern to us here, so we'll just call them groups. We'll describe the following groups: diplomonids, parabosilids, euglenozoans, alveolates, straminopols, red algae, chlorophytes, and choanoflagellates. Table 1, Figure 5. As we shall see, some of these protist clades consist of organisms with very diverse body plans. On the other hand, certain body plans, such as those of amoebas and those of slime molds, have arisen again and again during evolution in groups only distantly related to one another. We will begin our tour of protist clades with two of apparently ancient origin. Diplomonids and parabosilids. Two clades, the diplomonids and the parabosilids, appear to represent the earliest surviving branches in today's tree of eukaryotic life. It is likely that other clades diverged even earlier than the diplomonids and parabosilids. However, any such clades either were lost because of massive changes in the environment or remained hidden in rarely studied environments. Both the diplomonids and the parabosilids are unicellular organisms that lack mitochondria. This absence of mitochondria may be a derived condition. Ancestors of these organisms may have possessed mitochondria that were lost in the course of evolution. The existence of such organisms today shows that eukaryotic life is feasible without mitochondria, and for that reason, the diplomonids and paraphospholids are the focus of much attention. Giardia lamblia, a diplomonid, is a familiar parasite that contaminates water supplies and causes the intestinal disease giardiasis. This tiny organism has no mitochondria, chloroplasts, or other membrane-enclosed organelles, but it contains two nuclei bounded by nuclear envelopes, and it has a cytoskeleton and multiple flagella. Trichomonas vaginalis is a paraphospholid responsible for sexually transmitted disease in humans. Infection of the male urethra, where it may occur without symptoms, is less common than infection of the vagina. In addition to flagella, the paraphospholid have undulating membranes that also contribute to the cell's locomotion. Euglenozoans. The euglenozoans are clade of flagellates, unicellular organisms with flagella. They reproduce asexually by binary fission. There are two subgroups of euglenozoans, euglenoids and kinetoplastids. Euglenoids have anterior flagella. The euglenoids possess flagella rising from a pocket at the anterior end of the cell. Many members of the group are photosynthetic. Euglenoids used to be claimed by the zoologists as animals and by the botanists as plants. Like most other euglenoids, this common freshwater organism has a complex cell structure. It propels itself through the water with the longer of its two flagella, which may also serve as an anchor to hold the organism in place. The flagellum provides power by means of a wavy motion that spreads from base to tip. The second flagellum is often rudimentary. Euglena has very flexible nutritional requirements. Many species are always heterotrophic. Other species are fully autotrophic in sunlight, using chloroplasts to synthesize organic compounds through photosynthesis. The chloroplasts of euglenas are surrounded by three membranes. When kept in the dark, these euglenas lose their photosynthetic pigment and begin to feed exclusively on dead organic material floating in the water around them. Such a bleached euglena resynthesizes its photosynthetic pigment when it is returned to the light and becomes autotrophic again. But euglena cells treated with certain antibiotics or mutagens lose their photosynthetic pigment completely. Neither they nor their descendants are ever autotrophs again. However, those descendants function well as heterotrophs. Kinetoplastids have mitochondria that edit their own RNA. The kinetoplastids are unicellular, parasitic flagellates with a single, large mitochondrion. That mitochondrion contains a kinetoplast, a unique structure housing multiple, circular DNA molecules and associated proteins. Some of these DNA molecules encode guides that edit RNA within the mitochondrion. Some kinetoplastids are human pathogens. Sleeping sickness, one of the most dreaded diseases of Africa, is caused by the parasitic kinetoplastic trypanosoma. An insect, the tsetse fly, is a vector intermediate host of trypanosoma. Carrying its deadly cargo, the tsetse fly bites livestock, wild animals, and humans, infecting them with the parasite. Trypanosoma then multiplies in the mammalian bloodstream and produces toxins. When these parasites invade the nervous system, the neurological symptoms of sleeping sickness appear and are followed by death. 500,000 people now have sleeping sickness, and 80% of them will die of the disease. About 3 million headed livestock die from trypanosoma infections each year. Other trypanosomes cause leishmaniasis, Chagas disease, and East Coast fever, all are major diseases in the tropics. Alveolates. The alveolates are a clade of unicellular organisms. The shared derived trait synapomorphy that characterizes them is the possession of cavities called alveoli just below their plasma membranes. They are diverse in body form. The alveolate groups we'll consider here are the dinoflagellates, apicomplexans, and ciliates. Dinoflagellates are unicellular marine organisms with two flagella. The dinoflagellates are all unicellular, and most are marine organisms. A distinctive mixture of photosynthetic and accessory pigments gives their chloroplasts a golden brown color. The dinoflagellates are of great ecological, evolutionary, and morphological interest. They are among the most important primary photosynthetic producers of organic matter in the oceans. Many dinoflagellates are endosymbionts living within the cells of other organisms, including various invertebrates and even other marine protists. Dinoflagellates are particularly common endosymbionts in corals, to whose growth they contribute by photosynthesis. As we will see later in this chapter, endosymbiotic events have given rise to dinoflagellates with different numbers of membranes surrounding their chloroplasts. Some dinoflagellates are non-photosynthetic and live as parasites within other marine organisms. Dinoflagellates have a distinctive appearance. They generally have two flagella, one in an equatorial groove around the cell, the other starting at the same point as the first and passing down a longitudinal groove before extending into the surrounding medium. Some dinoflagellates, notably P. stereophysidae, take on different forms, including amoeboid ones, depending on environmental conditions. It has been claimed that P. can occur in at least 2 dozen distinct forms, although this claim is controversial. In any case, this remarkable dinoflagellate is highly toxic to fish and can, when present in great numbers, both stun and feed on them. Some dinoflagellates reproduce in enormous numbers in warm and somewhat stagnant waters. The result can be a red tide, so called because of the reddish color of the sea that results from the pigments of the dinoflagellates. During a red tide, the concentration of dinoflagellates may reach 60 million cells per liter of ocean water. Pfiesteria and certain other red tide species produce a potent nerve toxin that can kill tons of fish. The genus Deinonychus produces a toxin that can accumulate in shellfish in amounts that, although not fatal to the shellfish, may kill a person who eats the shellfish. Many dinoflagellates are bioluminescent. In complete darkness, cultures of these organisms emit a faint glow. If air is suddenly stirred or bubbled through the culture, the organisms each emit numerous bright flashes. A ship passing through a tropical ocean that contains a rich growth of these species produces a bow wave and wake that glow nearly as billions of these dinoflagellates discharge their light systems. Apicomplexans are parasites with unusual spores. Exclusively parasitic organisms, the apicomplexans derive their name from the apical complex, a mass of organelles contained within the apical end of their spores. These organelles help the apicomplexans spore invade its host tissues. Unlike many other protists, apicomplexans lack contractile vacuoles. Apicomplexans generally have an amorphous amoeboid body form. This body form has evolved over and over again in parasitic protists. It appears even among parasitic dinoflagellates, a group of organisms whose non-parasitic relatives, as we have just seen, have highly distinctive, complex body forms. Like many obligate parasites, apicomplexans have elaborate life cycles featuring asexual and sexual reproduction by a series of very dissimilar life stages. Often these stages are associated with two different types of host organisms. Toxoplasma, a genus of apicomplexans, causes opportunistic infections in AIDS patients. There are other pathogenic apicomplexans as well. The best known are the malarial parasites of the genus Plasmodium, a highly specialized group of organisms that spend part of their life cycle within human red blood cells, figure 6. Although it has been almost eliminated from the United States, malaria continues to be a serious problem in many tropical countries, as we saw at the beginning of this chapter. In terms of the number of people infected, malaria is one of the world's 3 most serious diseases, and it kills more than 1 million people each year. Female mosquitoes of the genus Anopheles transmit Plasmodium to humans. The parasite enters the human circulatory system when an infected Anopheles mosquito penetrates the human skin in search of blood. The parasites find their way to cells in the liver and the lymphatic system, change their form, multiply, and re-enter the bloodstream, attacking red blood cells. The apical complex enables Plasmodium to enter human liver and red blood cells. The parasites multiply inside red blood cells, which then burst, releasing new swarms of parasites. If another Anopheles bites the victim, the mosquito takes in Plasmodium cells along with blood. Some of these cells develop into gametes, which unite to form zygotes that lodge in the mosquito's gut, divide several times, and move into its salivary glands, from which they can be passed on to another human host. Thus, Plasmodium is an extracellular parasite in the mosquito vector and an intracellular parasite in the human host. Plasmodium has proved to be a singularly difficult pathogen to attack. The Plasmodium life cycle is best broken by the removal of stagnant water, in which mosquitoes breed. The use of insecticides to reduce the Anopheles population can be effective, but their benefits must be weighed against the possible ecological, economic, and health risks posed by the insecticides themselves. New hope arises from the publication, in 2002, of the genomes of both Plasmodium falciparum and one of its vectors, Anopheles gambiae, as well as a partial proteome of P. falciparum. These advances should lead to a better understanding of the biology of malaria and to the possible development of drugs, vaccines, or other means of dealing with this protest pathogen or its insect vectors. Ciliates have 2 types of nuclei. The ciliates are so named because they characteristically have hair-like cilia. This group is noteworthy for its diversity and ecological importance. Almost all ciliates are heterotrophic, a few contain photosynthetic endosymbionts, and they are much more complex in body form than are most flagellates and other unicellular protists. The definitive characteristic of ciliates is the possession of 2 types of nuclei, commonly a single macronucleus within the same cell, from 1 to several micronuclei. The micronuclei, which are typically eukaryotic nuclei, are essential for genetic recombination. The macronucleus is derived from micronuclei. Each macronucleus contains many copies of the genetic information, packaged in units containing very few genes each. The macronuclear DNA is transcribed and translated to regulate the life of the cell. Although we do not know how the system of macro and micronuclei came into being, we do know something about the behavior of these nuclei, which we will discuss after describing the body plan of one important ciliate, Paramecium. A closer look at one ciliate. Paramecium, a frequently studied ciliate genus, exemplifies the complex structure and behavior of ciliates, figure 7. The slipper-shaped cell is covered by an elaborate pellicle, a structure composed principally of an outer membrane and an inner layer of closely packed, membrane-enclosed sacs, the alveoli, that surround the bases of the cilia. Defensive organelles called trichocysts are also present in the pellicle. In response to a threat, a microscopic explosion expels the trichocysts in a few milliseconds, and they emerge as sharp darts, driven forward at the tip of a long, expanding filament. The cilia provide a form of locomotion that is generally more precise than locomotion by flagella or pseudopods. A paramecium can direct the beating of its cilia to propel itself either forward or backward in a spiraling manner. It can also back off swiftly when it encounters a barrier or a negative stimulus. The coordination of ciliary beating is probably the result of a differential distribution of ion channels in the plasma membrane near the 2 ends of the cell. Reproduction without sex and sex without reproduction. Paramecia reproduce mechanism from a round of DNA replication. Paramecia also have an elaborate sexual behavior called conjugation, in which two paramecia line up tightly against each other and fuse in the oral region of the body. Nuclear material is extensively reorganized and exchanged over the next several hours. Figure 8. As a result of this process, each cell ends up with two haploid micronuclei, one of its own and one from the other cell, which fuse to form a new diploid micronucleus. New macronuclei develop from the micronuclei through a series of dramatic chromosomal rearrangements. The exchange of nuclei is fully reciprocal. Each of the two paramecia gives and receives an equal amount of DNA. The two organisms then separate and go their own ways, each equipped with new combinations of alleles. Conjugation in paramecium is a sexual process of genetic recombination, but it is not a reproductive process. The same two cells that begin the process are there at the end, and no new cells are created. As a rule, each asexual clone of paramecium must periodically conjugate. Experiments have shown that if some species are not permitted to conjugate, the clones can live through no more than approximately 350 cell divisions before they die out. Stramenopels. The shared trait that defines the stramenopels is the possession of two flagella, typically unequal in length. The longer of the two bears rows of tubular hairs. Some stramenopels lack flagella, but they are presumed to be descended from ancestors that possess flagella. The stramenopels include the diatoms and the brown algae, which are photosynthetic, and the oomycetes, which are not. Other stramenopel groups include some lineages that are non-photosynthetic. Most golden algae are photosynthetic, but nearly all of them become heterotrophic when light intensity is limiting or when there is a plentiful food supply. Some even feed on diatoms or bacteria. Some botanists refer to the stramenopels as the brown plant kingdom. Diatoms are everywhere in the marine environment. Diatoms are single-celled organisms, although some species associate in filaments. Many have sufficient carotenoids in their chloroplasts to give them a yellow or brownish color. All make chrysolaminarin, a carbohydrate, and oils as photosynthetic storage products. Diatoms lack flagella. Architectural magnificence on a microscopic scale is a hallmark of the diatoms. Many diatoms deposit silicon in their cell walls. The cell wall of some species is constructed in two pieces, with the top overlapping the bottom like the top and bottom of a petri plate. The silicon-impregnated walls have intricate, unique patterns. Despite their remarkable morphological diversity, however, all diatoms are symmetrical, either bilaterally with right and left halves or radially with the type of symmetry possessed by a circle. Why are diatom cell walls so glassy and complex, and why are diatoms so abundant? A 2003 paper by German biologists may shed light on these questions. By measuring, at a microscopic scale, the forces needed to break single, living diatoms, the biologists discovered that their cell walls are exceptionally strong. Evolution of these walls by natural selection has given these diatoms an enhanced defense against predators and, thus, an edge over competitors. Diatoms reproduce both sexually and asexually. Asexual reproduction is by binary fission and is somewhat constrained by the stiff, silica-containing cell wall. Both the top and the bottom of the petri plate become tops of new plates without changing appreciably in size. As a result, the new cell made from the former bottom is smaller than the parent cell. Figure 9. If this process continued indefinitely, one cell line would simply vanish, but sexual reproduction largely solves this potential problem. Gametes are formed, shed their cell walls, and fuse. The resulting zygote then increases substantially in size before a new cell wall is laid down. Diatoms are everywhere in the marine environment and are frequently present in great numbers, making them major photosynthetic producers in coastal waters. Diatoms are also common in fresh water. Because the silicon-containing walls of dead diatom cells resist decomposition, certain sedimentary rocks are composed almost entirely of diatom skeletons that sink to the seafloor over time. Diatomaceous earth, which is obtained from such rocks, has many industrial uses, such as insulation, filtration, and metal polishing. It has also been used as an earth-friendly insecticide that clogs the trachea breathing structures of insects. The brown algae include the largest protists. All the brown algae are multicellular. They are composed either of branched filaments or of leaf-like growths called thallate, singular, thallus. The brown algae obtain their namesake color from the carotenoid fucoxanthin, which is abundant in the chloroplasts. The combination of this yellow-orange pigment with the green of chlorophylls and C yields a brownish tinge. The brown algae include the largest of the protists. Giant kelps, such as those at the genus Macrocystis, may be up to 16 meters long. The brown algae are almost exclusively marine. Some float in the open ocean. The most famous example is the genus Sargassum, which forms dense mats in the Sargasso Sea in the mid-Atlantic. Most brown algae, however, are attached to rocks near the shore. A few thrive only where they are regularly exposed to heavy surf. A notable example is the sea Compostelsia palmiformis of the Pacific coast. All of the attached forms develop a specialized structure, called a holdfast, that literally glues them to the rocks. Some brown algae differentiate extensively into stem-like stalks and leaf-like blades, and some develop gas-filled cavities or bladders. For biochemical reasons that are only poorly understood, these bladders often contain as much as 5% carbon monoxide, a concentration high enough to kill a human. In addition to organ differentiation, the larger brown algae also exhibit considerable tissue differentiation. Most of the giant kelps have photosynthetic filaments only in the outermost regions of their stalks and blades. Within these photosynthetic regions lie filaments of long cells that closely resemble the nutrient-conducting tissue of plants. Called trumpet cells because they have flaring ends, these tubes rapidly conduct the products of photosynthesis through the body of the organism. The cell walls of brown algae may contain as much as 25% alginic acid, a gummy polymer of sugar acids. Alginic acid cements cells and filaments together and provides good holdfast glue. It is used commercially as an emulsifier in ice cream, cosmetics, and other products. Many protists in all plant life cycles feature alternation of generations. Brown algae, like many other multicellular photosynthetic protists in all plants, exhibit a type of life cycle known as alternation of generations, in which a multicellular, diploid, spore-producing organism gives rise to a multicellular, haploid, gamete-producing organism. When two haploid gametes fuse, a process called fertilization, or syngamy, a diploid organism is formed. Figure 10. The haploid organism, the diploid organism, or both may also reproduce asexually. The two generations, spore-producing and gamete-producing, differ genetically. One has diploid cells and the other has haploid cells, but they may or may not differ morphologically. In heteromorphic alternation of generations, the two generations differ morphologically. In isomorphic alternation of generations, they do not, despite their genetic difference. We will see examples of both heteromorphic and isomorphic alternation of generations in some representative brown and green algae. In discussing the life cycles of plants and multicellular photosynthetic protists, we will use the term sporophyte, spore plant, and gametophyte, gamete plant, to refer to the multicellular diploid and haploid generations, respectively. Gametes are not produced by meiosis because the gametophyte generation is already haploid. Instead, specialized cells of the diploid sporophyte, called sporocytes, divide meiotically to produce 4 haploid spores. The spores may eventually germinate and divide meiotically to produce multicellular haploid gametophytes, which produce gametes by mitosis and cytokinesis. Gametes, unlike spores, can produce new organisms only by fusing with other gametes. The fusion of two gametes produces a diploid zygote, which then undergoes meiotic division to produce a diploid organism, the sporophyte generation. The sporocytes of the sporophyte generation then undergo meiosis and produce haploid spores, starting the cycle anew. The brown algae exemplify the extraordinary diversity found among the photosynthetic protists. One genus of simple brown algae is Ectocarpus. Its branched filaments, a few centimeters long, commonly grow on shells and stones. The gametophyte and sporophyte generations of Ectocarpus can be distinguished only by chromosome number or reproductive products, spores or gametes. Thus the generations are isomorphic. By contrast, some kelps of the genus Laminaria and some other brown algae show a more complex heteromorphic alternation of generations. The larger and more obvious generation of these species is the sporophyte. Meiosis in sporocytes located on the leaf-like fronds produces haploid zoospores, monospores that are propelled by flagella. These spores germinate to form a tiny, filamentous gametophyte that produces either eggs or sperm. The eggs and sperm of brown algae typically have flagella. The oomycetes include water molds and their relatives. A non-photosynthetic stramenopel group called the oomycetes consists in large part of the water molds and their terrestrial relatives, such as the downy mildews. Water molds are filamentous and stationary, and they feed by absorption, that is, they secrete enzymes that digest large food molecules into smaller molecules that the water molds can absorb. If you have seen a whitish, cotton mold grown on dead fish or dead insects in water, it was probably a water mold of the common genus Cipollina. Don't be confused by the mycete in the name. That term means fungus, and it is there because these organisms were once classified as fungi. However, we now know that the oomycetes are unrelated to the fungi. The oomycetes are coenocytes. They have many nuclei enclosed in a single plasma membrane. Their filaments have no cross walls to separate the many nuclei into discrete cells. Their cytoplasm is continuous throughout the body of the organism, and there is no single structural unit with a single nucleus, except in certain reproductive stages. A distinguishing feature of the oomycetes is their flagellated reproductive cells. Oomycetes are diploid throughout most of their life cycle and have cellulose in their cell walls. The water molds, such as Cipollina, are all aquatic and saprobic. They feed on dead organic matter. Some other oomycetes are terrestrial. Although most of the terrestrial oomycetes are harmless or helpful decomposers of dead matter, a few are serious plant parasites that attack crops such as avocados, grapes, and potatoes. The water mold Phytophthora infestans, for example, is a causal agent of late blight of potatoes, which brought about the Great Irish Potato Famine of 1845–1847. P. infestans destroyed the entire Irish potato crop in a matter of days in 1846. Among the consequences of the famine were a million deaths from starvation and the immigration of about 2 million people, mostly to the United States. Red algae. Almost all red algae are multicellular. Some plant biologists now refer to the red algae as the red plant kingdom. Their characteristic color is a result of the accessory photosynthetic pigment phycoerythrin, which is found in relatively large amounts in the chloroplasts of many species. In addition to phycoerythrin, red algae contain phycocyanin, carotenoids, and chlorophyll A. The red algae includes species that grow in the shallowest tide pools as well as the algae found deepest in the red algae includes species that grow in the shallowest tide pools as well as the algae found deepest in the ocean, as deep as 260 meters if nutrient conditions are right and the water is clear enough to permit the penetration of light. Very few red algae inhabit fresh water. The red algae includes species that grow in the shallowest tide pools as well as the algae found deepest in the ocean, as deep as 260 meters if nutrient conditions are right and the water is clear enough to permit the penetration of light. Very few red algae inhabit fresh water. Most grow attached to a substrate and bioholdfast. In a sense, the red algae, like several other groups of algae, are misnamed. They have the capacity to change the relative amounts of their various photosynthetic pigments depending on the light conditions where they are growing. Thus, the leaf-like Chondrus crispus, a common North Atlantic red alga, may appear bright green when it is growing near the surface of the water and deep red when growing at greater depths. The ratio of pigments present depends to a remarkable degree on the intensity of the light that reaches the alga. In deep water, where the light is dimmest, the alga accumulates large amounts of phycoerythrin. The algae in deep water have as much chlorophyll as the green ones near the surface, but the accumulated phycoerythrin makes them look red. In addition to being the only photosynthetic protists with phycoerythrin and phycocyanin among their pigments, the red algae have two other unique characteristics. First, they store the products of photosynthesis in the form of fluorine starch, which is composed of very small, branched chains of approximately 15 glucose units. Second, they produce no motile, flagellated cells at any stage in their life cycle. The male gametes lack cell walls and are slightly amoeboid. The female gametes are completely immobile. Some red algal species enhance the formation of coral reefs. Like coral animals, they possess the biochemical machinery for secreting calcium carbonate, which they deposit both in and around their cell walls. After the death of the corals and algae, the calcium carbonate persists, sometimes forming substantial rocky masses. Some red algae produce large amounts of mucilaginous polysaccharide substances, which contain the sugar galactose with a sulfate group attached. This material readily forms solid gels and is the source of agar, a substance widely used in the laboratory for making a solid aqueous medium on which tissue cultures and many microorganisms can be grown. Certain red algae became endosymbionts long ago within the cells of other, non-photosynthetic protists, eventually giving rise to chloroplasts. They are the ancestors of the distinctive chloroplasts of the photosynthetic straminopples, the brown algae and the diatoms. Chlorophytes. The green algae do not form a clade, but they include at least two multicellular clades. One major clade consists of the chlorophytes. A sister clade to the chlorophytes contains another green algal clade along with the plant kingdom. The green algal clades share characters that distinguish them from other protists. Like the plants, they contain chlorophylls and B, and a reserve of photosynthetic products is stored as starch in plastids. The chlorophytes are the largest clade of green algae, containing more than 17,000 species. Most are aquatic, some are marine, but more are freshwater forms, but others are terrestrial, living in moist environments. The chlorophytes range in size from microscopic unicellular forms to multicellular forms many centimeters in length. Chlorophytes vary in shape and cellular organization. We find among the chlorophytes an incredible variety in shape and construction of the algal body. Chlamydomonas is an example of the simplest type, unicellular and flagellated. Surprisingly large and well-formed colonies of cells are found in such freshwater groups as the genus Volvox. The cells in these colonies are not differentiated into tissues and organs, as in plants and animals, but the colonies show vividly how the preliminary step of this great evolutionary development might have been taken. In Volvox, the origins of cell specialization can be seen in certain cells within the colony that are specialized for reproduction. While Volvox is colonial and spherical, Oedogonium is multicellular and filamentous, and each of its cells has only one nucleus. Cladophora is multicellular, but each cell is multinucleate. Bryopsis is tubular and coenacidic, forming cross walls only when reproductive structures form. A setaviolaria is a single, giant uninucleate cell a few centimeters long that becomes multinucleate only at the end of its reproductive stage. Vulva-lactuca is a thin, membranous sheet a few centimeters across. Its unusual appearance justifies its common name, sea lettuce. Chlorophyte life cycles are diverse. The life cycles of chlorophytes show great diversity. Let's examine two chlorophyte life cycles in detail, beginning with that of the sea lettuce Vulva-lactuca, figure 11. Like many chlorophytes, sea lettuce exhibits alternation of generations. The diploid sporophyte of this common seashore organism is a broadsheet only two cells thick. Some of its cells, sporocytes, differentiate and undergo meiosis and cytokinesis, producing motile haploid spores, zoospores. These swim away, each propelled by four flagella, and some eventually find a suitable place to settle. The spores then lose their flagella and begin to divide mesotically, producing a thin filament that develops into a broadsheet only two cells thick. The gametophyte thus produced looks just like the sporophyte. In other words, Vulva-lactuca has an isomorphic life cycle. In Vulva-lactuca, an individual gametophyte can produce only male or female gametes, never both. The gametes arise mesotically within single cells called gametangia rather than within a specialized multicellular structure, as in plants. Both types of gametes bear two flagella in contrast to the four flagella of a haploid spore and hence are mobile. In most species of vulva, the female and male gametes are indistinguishable structurally, making those species isogamous, having gametes of identical appearance. Other chlorophytes, including some other species of vulva, are anisogamous, having female gametes that are distinctly larger than the male gametes. Female and male gametes come together and unite, losing their flagella as the zygote forms and settles. After resting briefly, the zygote begins meiotic division, producing a multicellular sporophyte. Any gametes that fail to find partners can settle down on a favorable substratum, lose their flagella, undergo mitosis, and produce a new gametophyte directly. In other words, the gametes can also function as zoospores. Few chlorophytes other than vulva have mobile gametes that can also function as zoospores. In contrast to the isomorphic life cycle of vulva, many other chlorophytes have a heteromorphic life cycle, in which sporophyte and gametophyte generations differ in structure. In one variation of the heteromorphic life cycle, the haplontic life cycle, figure 12, a multicellular haploid individual produces gametes that fuse to form a zygote. The zygote functions directly as a sporocyte, undergoing meiosis to produce spores, which in turn produce a new haploid individual. In the entire haplontic life cycle, only one cell, the zygote, is diploid. The filamentous organisms of the genus Euliphrix are examples of haplontic chlorophytes. Some other chlorophytes have a diplontic life cycle like that of many animals. In a diplontic life cycle, meiosis of diploid sporocytes produces haploid gametes directly. The gametes fuse, and the resulting diploid zygote divides mesotically to form a new multicellular sporophyte. In such organisms, every cell except the gametes is diploid. Between these two extremes are chlorophytes in which the gametophyte and sporophyte generations are both multicellular, but one generation, usually the sporophyte, is much larger and more prominent than the other. There are green algae other than chlorophytes. As we mentioned above, the chlorophytes are the largest clade of green algae, but there are other green algal clades as well. Those clades are branches of a clade that also includes the plant kingdom. The green algal clade that is sister to the plant kingdom, containing a group of organisms called charophytes, will be described in the next chapter. But now let's consider some close protist relatives of the animals. Choanoflagellates. One group of heterotrophic protists with flagella, the choanoflagellates, is thought to comprise the closest relatives of the animals. The choanoflagellates are sister to the animals, and the animal choanoflagellate lineage is sister to the fungi. The clade consisting of fungi, animals, and choanoflagellates is called the opisthokonts. Choanoflagellates are colonial and are thought to be closely related to the sponges, the most ancient of the surviving phyla of animals. Choanoflagellates bear a striking resemblance to the most characteristic type of cell found in the sponges. Sponges are also colonial rather than truly multicellular, in that they lack organized tissues and their cells can be separated and allowed to reaggregate. We turn now to a topic hinted at several times above, the history of unusual numbers of membranes surrounding the chloroplasts of some photosynthetic protists. A history of endosymbiosis. As we have already seen, many protists possess chloroplasts. Groups with chloroplasts appear in several distantly related protist clades. Some of these groups differ in the photosynthetic pigments their chloroplasts contain. And we've seen that not all chloroplasts have a pair of surrounding membranes. In some protists, they are surrounded by three membranes. We now understand these observations in terms of a remarkable series of endosymbiosis. All chloroplasts trace their ancestry back to the engulfment of a cyanobacterium by a larger eukaryotic cell, figure 13. This event is known as primary endosymbiosis. The cyanobacterium, a gram-negative bacterium, had both an inner and outer membrane. The eukaryote's plasma membrane wrapped around the cyanobacterium as it took it up. The outer membrane and cell wall of the cyanobacterium were eventually lost. Thus, the original chloroplast had two surrounding membranes, one from the cyanobacterium and one from the eukaryotic host cell. Primary endosymbiosis gave rise to the chloroplasts of the green algae and the red algae. We do not yet know whether both trace back to a single primary endosymbiosis, as is likely, with later divergence, or whether they resulted from independent occurrences of primary endosymbiosis. In either case, each line participated in further endosymbiosis. All remaining photosynthetic eukaryotes are the result of secondary or tertiary endosymbiosis. The photosynthetic euglenoids derive their chloroplasts from secondary endosymbiosis. Their ancestor took up a unicellular chlorophyte, retaining the endosymbiont chloroplast and eventually losing the rest of its constituents. This history accounts for the fact that the photosynthetic euglenoids have the same photosynthetic pigments as the chlorophytes in plants. It also accounts for the third membrane of the euglenoid chloroplast, which is derived from the euglenoid's plasma membrane. Other photosynthetic protist groups derive their chloroplasts by secondary endosymbiosis with unicellular red algae. Both the green clade and the red clade of chloroplasts appear to have been involved in more than one secondary endosymbiosis. At least one secondary endosymbiosis produced a unicellular protist that became, itself, a partner in a tertiary endosymbiosis. In that case, a dinoflagellate lost its plastid and took up a haptophyte protist, itself the result of secondary endosymbiosis. The result is a dinoflagellate Karenia brevis. Although euglenoid chloroplasts are descendants of the chlorophyte and straminople chloroplasts are descendants of the red alga, this does not mean that euglenoids themselves are descendants of the chlorophyte, nor are straminopoles themselves descendants of the red alga. The ancestors that took up green or red algae in secondary endosymbiosis had their own evolutionary histories. Thus, the nuclear and chloroplast genomes of these protists have different histories. It has taken much research to piece together the clades as we now understand them. The clades of protists that we have discussed are summarized in Table 1 and Figure 5. Now let's consider some of the body forms that have appeared repeatedly in various branches of the eukaryote family tree. Some recurrent body forms. Amoebas used to be classified together in a single protist group. We now know that the amoebas map to at least two complex assemblages that are difficult to position in phylogenetic trees. Similarly, three kinds of organisms called slime molds, once classified together, may be quite different phylogenetically. In this section, we look at some of the variations on these body plans found among the protists. Amoebas form pseudopods. The pseudopods used by amoebas for locomotion are a hallmark of the amoeboid body plan. This body plan has appeared by convergent evolution in various protist groups. Amoebas have often been portrayed in popular writing as blobs, the simplest form of animal life imaginable. Superficial examination of a typical amoeba shows how such an impression might have been obtained. An amoeba consists of a single cell. It feeds on small organisms and particles of organic matter by phagocytosis, engulfing them with its pseudopods. But amoebas are specialized protists. Many are adapted for life on the bottoms of lakes, ponds, and other bodies of water. Their creeping locomotion and a manner of engulfing food particles fit them for life close to a relatively rich supply of sedentary organisms or organic particles. Most amoebas exist as predators, parasites, or scavengers. A few are photosynthetic. Amoebas of the free-living genus Naegleria, some of which can enter humans and cause a fatal disease of the nervous system, have a two-stage life cycle, one stage having amoeboid cells and the other flagellated cells. Some amoebas are shelled, living in casings of sand grains glued together. Others have shells secreted by the organism itself. Actinopods have thin, stiff pseudopods. The actinopods are recognizable by their thin, stiff pseudopods, which are reinforced by microtubules. These pseudopods play at least four roles. They greatly increase the surface area of the cell for exchange of materials with the environment. They help the cell float in its marine or freshwater environment. They provide locomotion in some species. They are the cell's feeding organs, trapping smaller organisms and often taking them up by endocytosis. Radiolarians, a group of actinopods found exclusively in marine environments, are perhaps the most beautiful of all microorganisms. Almost all radiolarian species secrete glassy endoskeletons, internal skeletons from which needle-like pseudopods project. Part of the skeleton is a central capsule within the cytoplasm. The skeletons of the different species are as varied as snowflakes, and many have elaborate geometric designs. A few radiolarians are among the largest of the unicellular protists, measuring several millimeters across. Innumerable radiolarian skeletons, some as old as 700 million years, form the sediments under some tropical seas. Heliozoans are actinopods that lack an endoskeleton, figure 28.30b. Most heliozoans are found in freshwater. They roll along the substratum by shortening and elongating their pseudopods. Foraminiferanes have created vast limestone deposits. Foraminiferanes are marine protists that secrete shells of calcium carbonate. The shells of individual foraminiferae species have distinctive shapes. Some foraminiferanes live as plankton, free-floating microscopic organisms, and many others live at the bottom of the sea. Their long, thread-like, branched pseudopods reach out through numerous microscopic pores in the shell and interconnect to create a sticky net, which the foraminiferae uses to catch smaller plankton. After foraminiferanes reproduce by mitosis and cytokinesis, the daughter cells abandon the parent shell and make new shells of their own. The discarded skeletons of ancient foraminiferanes make up extensive limestone deposits in various parts of the world, forming a layer hundreds of thousands of meters deep over millions of square kilometers of ocean bottom. Foraminiferae skeletons also make up the sand of some beaches. A single gram of such sand may contain as many as 50,000 foraminiferae shells and shell fragments. The shells of individual foraminiferanes are easily preserved as fossils in marine sediments. Each geological period has distinctive foraminiferae species. For this reason, and because they are so abundant, the remains of foraminiferanes are especially valuable as indicators in the classification and dating of sedimentary rocks, as well as in oil prospecting. They also reveal information about temperatures at the time they were alive. Slime molds release spores from erect fruiting bodies. The three groups of slime molds seem so similar at first glance that they were once grouped together in a single phylum. However, the slime molds are actually so different that some biologists now classify them in different kingdoms. We will consider two of these groups, called acellular slime molds and cellular slime molds. The slime molds share only general characteristics. All are motile, all ingest particulate food by endocytosis, and all form spores on erect fruiting bodies. They undergo striking changes in organization during their life cycles, and one stage consists of isolated cells that take up food particles by endocytosis. Some slime molds may cover areas of 1 meter or more in diameter while in their less aggregated stage. Such a large slime mold may weigh more than 50 grams. Slime molds of both types favor cool, moist habitats, primarily in forests. They range from colorless to brilliantly yellow and orange. Acellular slime molds form multinucleate masses. If the nucleus of an amoeba began rapid meiotic division, accompanied by a tremendous increase in cytoplasm and organelles, the resulting organism might resemble the acellular slime molds myxomycetes. During its vegetative feeding phase, an acellular slime mold is a wall-less mass of cytoplasm with numerous diploid nuclei. This mass strings very slowly over its substratum in a remarkable network of strands called a plasmodium. The plasmodium of an acellular slime mold is another example of a coenocyte, a body in which many nuclei are enclosed in a single plasma membrane. The outer cytoplasm of the plasmodium closest to the environment is normally less fluid than the interior cytoplasm and thus provides some structural rigidity. Acellular slime molds, such as Physarum, provide a dramatic example of movement by cytoplasmic streaming. The outer cytoplasmic region of the plasmodium becomes more fluid in places, and cytoplasm rushes into those areas, stretching the plasmodium. This streaming somehow reverses its direction every few minutes as cytoplasm rushes into a new area and drains away from an older one, moving the plasmodium over its substratum. Sometimes an entire wave of plasmodium moves across the substratum, leaving strands behind. Actin filaments and a contractile protein called mesomyosin interact to produce a streaming movement. As it moves, the plasmodium engulfs food particles by endocytosis, predominantly bacteria, yeast, spores of fungi, and other small organisms, as well as decaying animal and plant remains. An acellular slime mold can grow almost indefinitely in its plasmodial stage, as long as the food supply is adequate and other conditions, such as moisture and pH, are favorable. However, one of two things can happen if conditions become unfavorable. First, the plasmodium can form an irregular mass of hardened cell-like components called a sclerotium. This resting structure rapidly becomes a plasmodium again when favorable conditions are restored. Alternatively, the plasmodium can transform itself into spore-bearing fruiting structures. These stalked or branched structures, called sporangiophores, rise from heaped masses of plasmodium. They derive their rigidity from walls that form and thicken between their nuclei. The nuclei of the plasmodium are diploid, and they divide by meiosis as the sporangiophore develops. One or more knobs, called sporangia, develop on the end of the stalk. Within a sporangium, haploid nuclei become surrounded by walls and form spores. Eventually, as the sporangiophore dries, it sheds its spores. The spores germinate into wall-less, flagellated, haploid cells called swarm cells, which can either divide nucleoidally to produce more haploid swarm cells or function as gametes. Swarm cells can live as separate individual cells and can become walled and resistant resting cysts when conditions are unfavorable. When conditions improve again, the cysts release flagellated swarm cells. Two swarm cells can also fuse to form a diploid zygote, which divides by mitosis but without a wall forming between the nuclei and thus forms a new, coenecytic plasmodium. Cells retain their identity in the cellular slime molds. Whereas the plasmodium is the basic vegetative unit of the acellular slime molds, an amoeboid cell is the vegetative unit of the cellular slime molds. Large numbers of cells called myxamoebas, which have single haploid nuclei, engulf bacteria and other food particles by endocytosis and reproduce by mitosis and fission. This simple life cycle stage, consisting of swarms of independent, isolated cells, can persist indefinitely as long as food and moisture are available. When conditions become unfavorable, however, the cellular slime molds aggregate and form fruiting structures, as do their acellular counterparts. The apparently independent myxamoebas aggregate into a mass called a slug or pseudoplasmodium, figure 14. Unlike the true plasmodium of the acellular slime molds, this structure is not simply a giant sheet of cytoplasm with many nuclei. The individual myxamoebas retain their plasma membranes and, therefore, their identity. The chemical signal that causes the myxamoebas of cellular slime molds to aggregate into a slug is 3 feet, 5 feet cyclic adenosine monophosphate, a compound that plays many important roles as a chemical signal in animals. A slug may migrate over its substratum for several hours before becoming motionless and reorganizing to construct a delicate, stocked fruiting structure, figure 14. Cells at the top of the fruiting structure develop into thick-walled spores, which are eventually released. Later, under favorable conditions, the spores germinate, releasing myxamoebas. The cycle from myxamoebas through slug and spores to new myxamoebas is asexual. Cellular slime molds also have a sexual cycle, in which two myxamoebas fuse. The product of this fusion develops into a spherical structure that ultimately germinates, releasing new haploid myxamoebas. This asexual life cycle, individual cells forming about and then aggregating to form a fruiting body, has evolved independently more than once. A bacterial group, the myxobacteria, also has swarming cells that aggregate and form fruiting structures. These bacteria are, of course, prokaryotes and thus have no sexual cycle. The plant kingdom. The kingdom Plantae is monophyletic. All plants descend from a single common ancestor and form a branch of the evolutionary tree of life. The shared derived trait, or synapomorphy, of the plant kingdom is development from embryos protected by tissues of the parent plant. For this reason, plants are sometimes referred to as embryophytes. Plants retain the derived features that they share with green algae, the use of chlorophylls and the use of starch as a photosynthetic storage product. Both plants and green algae have cellulose in their cell walls. There are other ways to define plant and plant kingdom and still come out with a monophyletic group clade. For example, combining plants is defined above with a group of green algae called the charophytes results in a monophyletic plant kingdom with several shared derived traits, including the retention of the egg in the parent body. The addition of the chlorophytes, the remainder of the green algae, to the group just described gives another monophyletic group, with synapomorphies including the possession of chlorophyll B, that can be called the plant kingdom. There are no hard and fast criteria for defining a kingdom or any other taxonomic rank, so these definitions of the plant kingdom are all valid. Figure 1. There are 10 surviving phyla of plants. The surviving members of the kingdom plant fall naturally into 10 phyla. Table 1. All members of 7 of those phyla possess well-developed vascular systems that transport materials throughout the plant body. We call these 7 phyla, collectively, the tracheophytes because they all possess conducting cells called trachees. The tracheophytes constitute a clade. The remaining 3 phyla, liverworts, hornworts, and mosses which lack trachees, were once considered classes of a single larger phylum. In this book we use the term non-tracheophytes to refer collectively to these 3 phyla. The non-tracheophytes are sometimes collectively called bryophytes, but in this text we reserve that term for the most familiar members, the mosses. Collectively, the non-tracheophytes are not a monophyletic group. They are the 3 basal clades of the plant kingdom. Life cycles of plants feature alternation of generations. A universal feature of the life cycles of plants is the alternation of generations. Alternation of generations has 2 hallmarks. The life cycle includes both multicellular diploid individuals and multicellular haploid individuals. Gametes are produced by mitosis, not by meiosis. Meiosis produces spores that develop into multicellular haploid individuals. If we begin looking at the plant life cycle at a single cell stage, the diploid zygote, then the first phase of the cycle features the formation, by mitosis and cytokinesis, of a multicellular embryo and eventually the mature diploid plant. Figure 2. This multicellular, diploid plant is a sporophyte, spore plant. Cells contained in sporangia singular, sporangium, spore vessel, on the sporophyte undergo meiosis to produce haploid, unicellular spores. By mitosis and cytokinesis, a spore forms a haploid plant. This multicellular, haploid plant is a gametophyte gamete plant that produces haploid gametes. The fusion of 2 gametes, syngamy, or fertilization results in the formation of a diploid cell, the zygote, and the cycle repeats. The sporophyte generation extends from the zygote through the adult, multicellular, diploid plant. The gametophyte generation extends from the spore through the adult, multicellular, haploid plant to the gamete. The transitions between the generations are accomplished by fertilization and meiosis. In all plants, the sporophyte and the gametophyte differ genetically. The sporophyte has diploid cells, and the gametophyte has haploid cells. In the 3 basal plant clades, the gametophyte generation is larger and more self-sufficient, while the sporophyte generation is dominant in those groups that appeared later in plant evolution. Some protist life cycles also feature alternation of generations, suggesting that the plants arose from one of these protist groups. But which one? The plant arose from a green algal clade. Much evidence indicates that the closest living relatives of the plants are members of a clade of green algae called the charophytes. The charophytes, along with some other green algae in the plants, form a clade that is sister to the chlorophytes. See figure 1, but we don't yet know which charophyte clade is the true sister group to the plants. Stoneworts of the genus Chara are charophytes that resemble plants in terms of their RNA and DNA sequences, peroxisome contents, mechanics of mitosis and cytokinesis, and chloroplast structure. On the other hand, strong evidence from morphology-based holistic analysis suggests that the sister group of the plants is a group of charophytes that includes the genus Coleoptete. Coleoptete-like algae have several features found in plants, such as plasmodesmata and a tendency to protect the young sporophyte. Whether they were more similar to stoneworts or to Coleoptete, the ancestors of the plants lived at the margins of ponds or marshes, ridding them with a green mat. From these marginal habitats, which were sometimes wet and sometimes dry, early plants made the transition onto land. The conquest of the land. Plants, or their immediate ancestors in the green mat, first invaded the terrestrial environment between 400 and 500 million years ago. That environment differs dramatically from the aquatic environment. The most obvious difference is the availability of the water that is essential for life. It is everywhere in the aquatic environment, but hard to find and to retain in the terrestrial environment. Water provides aquatic organisms with support against gravity. A plant on land, however, must either have some other support system or sprawl and support it on the ground. A land plant must also use different mechanisms for dispersing its gametes and progeny than its aquatic relatives, which can simply release them into the water. How did organisms descended from aquatic ancestors adapt to such a challenging environment? Adaptations to life on land distinguish plants from green algae. Most of the characteristics that distinguish plants from green algae are evolutionary adaptations to life on land. Several of these features probably evolved in the common ancestor of the plants. The cuticle, a waxy covering that retards desiccation, drying. Gametangia, cases that enclose plant gametes and prevent them from drying out. Embryos, which are young sporophytes contained within a protective structure. Certain pigments that afford protection against the mutagenic ultraviolet radiation that bathes the terrestrial environment. Thick spore walls containing a polymer that protects the spores from desiccation and resists decay. A mutualistic association with a fungus that promotes nutrient uptake from the soil. Further adaptations to the terrestrial environment appear as plants continue to evolve. One of the most important of these later adaptations was the appearance of vascular tissues. Most present-day plants have vascular tissues. The first plants were non-vascular, lacking both water conducting and food conducting tissue. Although the term non-vascular plants is a time-honored name, it is misleading when applied to the entire non-tracheophyte group, because some mosses, unlike liverworts and hornworts, do have a limited amount of simple conducting tissue. Thus, the more unwieldy name, non-tracheophyte, is more descriptive. The first true tracheophytes, possessing specialized conducting cells called trachees, arose later. Figure 3. The non-tracheophytes, the liverworts, hornworts, and mosses have never been large plants. Except for some of the mosses, they have no water conducting tissue, yet some are found in dry environments. Many grow in dense masses, through which water can move by capillary action. Non-tracheophytes also have leaf-like structures that rarely catch and hold any water that splashes onto them. These plants are small enough that minerals can be distributed throughout their bodies by diffusion. Familiar tracheophytes include the club mosses, ferns, conifers, and angiosperms flowering plants. Tracheophytes differ from liverworts, hornworts, and mosses in crucial ways, one of which is the possession of a well-developed vascular system consisting of specialized tissues for the transport of materials from one part of the plant to another. One such tissue, the phloem, conducts the products of photosynthesis from sites where they are produced or released to sites where they are used or stored. The other vascular tissue, the xylem, conducts water and minerals from the soil to aerial parts of the plant. Because some of its cell walls are stiffened by a substance called lignin, xylem also provides support in the terrestrial environment. Non-tracheophyte plants evolved tens of millions of years before the earliest tracheophytes, even though tracheophytes appear earlier in the fossil record. The oldest tracheophyte fossils date back more than 410 million years, whereas the oldest non-tracheophyte fossils are only about 350 million years old, dating from a time when tracheophytes were already widely distributed. This finding simply shows that, given the differences in their structures and the chemical makeup of their cell walls, tracheophytes are more likely to form fossils than non-tracheophytes are. The non-tracheophytes, liverworts, hornworts, and mosses. Most liverworts, hornworts, and mosses grow in dense mats, usually in moist habitats. The largest of these plants are only about 1 meter tall, and most are only a few centimeters tall along. Why have the non-tracheophytes not evolved to be taller? The probable answer is that they lack an efficient system for conducting water and minerals from the soil to distant parts of the plant body. To limit water loss, layers of maternal tissue protect the embryos of all non-tracheophytes. All non-tracheophyte clades also have a cuticle, although it is often very thin or even absent in some species and thus not highly effective in retarding water loss. Non-tracheophytes lack the leaves, stems, and roots that characterize tracheophytes, although they have structures analogous to each. Most non-tracheophytes live on the soil or on other plants, but some grow on bare rock, dead and fallen tree trunks, and even on buildings. Non-tracheophytes are widely distributed over 6 continents and exist very locally on the coast of the 7th Antarctica. They are successful plants, well adapted to their environments. Most are terrestrial, some live in wetlands. Although a few non-tracheophyte species live in freshwater, these aquatic forms are descended from terrestrial ones. There are no marine non-tracheophytes. Non-tracheophyte sporophytes are dependent on gametophytes. In non-tracheophytes, the conspicuous green structure visible to the naked eye is the gametophyte. Figure 4. In contrast, the familiar forms of tracheophytes, such as ferns and sea plants, are sporophytes. The gametophyte of non-tracheophytes is photosynthetic and therefore nutritionally independent, whereas the sporophyte may or may not be photosynthetic, but is always nutritionally dependent on the gametophyte and remains permanently attached to it. A non-tracheophyte sporophyte produces unicellular, haploid spores as products of meiosis within a sporangium, or capsule. A spore germinates, giving rise to a multicellular, haploid gametophyte whose cells contain chloroplasts and are thus photosynthetic. Eventually gametes form within specialized sex organs, the gametangia. The archegonium is a multicellular, flask-shaped female sex organ with a long neck and a swollen base, which produces a single egg. Figure 5a. The antheridium is a male sex organ in which sperm, each bearing 2 flagella, are produced in large numbers. Figure 5b. Once released, the sperm must swim or be splashed by raindrops to a nearby archegonium on the same or a neighboring plant. The sperm are aided in this task by chemical attractants released by the egg or the archegonium. Before sperm can enter the archegonium, certain cells in the neck of the archegonium must break down, leaving a water-filled canal through which the sperm swim to complete their journey. Note that all of these events require liquid water. On arrival at the egg, the nucleus of the sperm fuses with the egg nucleus to form a zygote. Meiotic divisions of the zygote produce a multicellular, diploid sporophyte embryo. The base of the archegonium grows to protect the embryo during its early development. Eventually, the developing sporophyte elongates sufficiently to break out of the archegonium, but it remains connected to the gametophyte by a foot that is embedded in the parent tissue and absorbs water and nutrients from it. The sporophyte remains attached to the gametophyte throughout its life. The sporophyte produces a capsule, within which meiotic divisions produce spores and thus the next gametophyte generation. The structure and pattern of elongation of the sporophyte differ among the 3 non-tracheophyte phyla, the liverworts, hepatophyta, hornworts, and the hornworts, anthocerophyta, and mosses, bryophyta. The probable evolutionary relationships of these 3 phyla and the tracheophytes can be seen in figure 3. Liverworts may be the most ancient surviving plant clade. The gametophytes of some liverworts, phylum Hepatophyta, are green, leaf-like layers that lie flat on the ground. Figure 29.7. The simplest liverwort gametophytes, however, are flat plates of cells, a centimeter or so long, that produce antheridial archegonia on their upper surfaces and anchoring water-absorbing filaments called rhizoids on their lower surfaces. Liverwort sporephytes are shorter than those of mosses and hornworts, rarely exceeding a few millimeters. The liverwort sporephyte has a stalk that connects capsule and foot. In most species, the stalk elongates and thus raises the capsule above ground level, favoring dispersal of spores when they are released. The capsules of liverworts are simple, a globular capsule wall surrounding a mass of spores. In some species of liverworts, spores are not released by the sporephyte until the surrounding capsule wall rots. In other liverworts, however, the spores are thrown from the capsule by structures that shorten and compress a spring as they dry out. When the stress becomes sufficient, the compressed spring snaps back to its resting position, throwing spores in all directions. Among the most familiar liverworts are species of the genus Marchantia, figure 6a. Marchantia is easily recognized by the characteristic structures on which its male and female gametophytes bear their antheridia and archegonia, figure 6b. Like most liverworts, Marchantia also reproduces asexually by simple fragmentation of the gametophyte. Marchantia and some other liverworts and mosses also reproduce asexually by means of gemmae singular, gemma, which are lens-shaped clumps of cells. In a few liverworts, the gemmae are loosely held in structures called gemmae cups, which promote dispersal of the gemmae by raindrops, figure 6c. Hornworts evolved stomata as an adaptation to terrestrial life. The phylum Anthoceratophyta comprises the hornworts, so named because their sporephytes look like little horns, figure 7. Hornworts appear at first glance to be liverworts with very simple gametophytes. These gametophytes consist of flat plates of cells with few cells thick. However, the hornworts, along with the mosses and tracheophytes, share an advance over the liverwort clade in their adaptation to life on land. They have stomata, pores that, when open, allow the uptake of CO2 for photosynthesis and the release of O2. Stomata may be a shared derived trait synapomorphy of hornworts and all other plants except liverworts, although hornworts stomata do not close and may have evolved independently. Hornworts have two characteristics that distinguish them from both liverworts and mosses. First, the cells of hornworts each contain a single large, plate-like chloroplast, whereas the cells of other non-tracheophytes contain numerous small, lens-shaped chloroplasts. Second, of all the non-tracheophyte sporephytes, those of the hornworts come closest to being capable of indeterminate growth without a set limit. Liverwort and moss sporephytes have a stalk that stops growing as the capsule matures, so elongation of the sporephyte is strictly limited. The hornwort sporephyte, however, has no stalk. Instead, a basal region of the capsule remains capable of indefinite cell division, continuously producing new spore-bearing tissue above. The sporephytes of some hornworts growing in mild and continuously moist conditions can become as tall as 20 centimeters. Eventually, the sporephyte's growth is limited by the lack of a transport system. To support their metabolism, the hornworts need access to nitrogen. Hornworts have internal cavities filled with mucilage. These cavities are often populated by cyanobacteria that convert atmospheric nitrogen gas into a form usable by the host plant. We have presented the hornworts as sister to the clade consisting of mosses and tracheophytes, but this is only one possible interpretation of the current data. The exact evolutionary status of the hornworts is still unclear, and in some phylogenetic analyses they are placed as the most ancient plant clade. Water and sugar transport mechanisms emerged in the mosses. The most familiar non-tracheophytes are the mosses phylum Bryophyta. There are more species of mosses than of liverworts and hornworts combined, and these hardy little plants are found in almost every terrestrial environment. They are often found on damp, cool ground, where they form thick mats, figure 29.9. The mosses are probably sister to the rachioophytes, see figure 29.4. Many mosses contain a type of cell called a hydroid, which dies and leaves a tiny channel through which water can travel. The hydroid may be the progenitor of the trachid, the characteristic water-conducting cell of the tracheophytes, but it lacks lignin, a waterproofing substance that also lends structural support, and the cell wall structure found in trachetes. The possession of hydroids and a limited system for transport of sucrose by some mosses via cells called leptoids shows that the old term, non-vascular plant, is somewhat misleading when applied to mosses. In contrast to liverworts and hornworts, the sporephytes of mosses and tracheophytes grow by apical cell division, in which a region of the growing tip provides an organized pattern of cell division, elongation, and differentiation. This growth pattern allows extensive and sturdy vertical growth of sporephytes. Apical cell division is a shared derived trait of mosses and tracheophytes. The moss gametophyte that develops following spore germination is a branched, filamentous structure called the protonema, see figure 4. Although the protonema looks a bit like a filamentous green alga, it is unique to the mosses. Some of the filaments contain chloroplasts and are photosynthetic, others, called rhizoids, are non-photosynthetic and anchor the protonema to the substratum. After a period of linear growth, cells close to the tips of the photosynthetic filaments divide rapidly in three dimensions to form buds. The buds eventually differentiate a distinct tip, or apex, and produce the familiar leafy moss shoot with leaf-like structures arranged spirally. These leafy shoots produce antheridia or archegonia, see figure 5. The antheridia release sperm that travel through liquid water to the archegonia, where they fertilize the eggs. Sporophyte development in most mosses follows a precise pattern, resulting ultimately in the formation of an absorptive foot anchor to the gametophyte, a stalk, and, at the tip, a swollen capsule, the sporangium. In contrast to hornworts, whose sporophytes grow from the base, the moss sporophyte stalk grows at its apical end, as tracheophytes do. Cells at the tip of the stalk divide, supporting the elongation of the structure and giving rise to the capsule. For a while, the archegonial tissue grows rapidly as the stalk elongates, but eventually the archegonium is outgrown and is torn apart by the expanding sporophyte. The lid of the capsule is shed after the completion of meiosis and spore development. In most mosses, groups of cells just below the lid form a series of tube-like structures surrounding the opening. Highly responsive to humidity, these structures dig into the mass of spores when the atmosphere is dry. Then, when the atmosphere becomes moist, they fling out, scooping out the spores as they go, figure B. The spores are thus dispersed when the surrounding air is moist, that is, when conditions favor their subsequent germination. Mosses of the genus Sphagnum often grow in swampy places, where the plants begin to decompose in the water after they die. Rapidly growing upper layers compress the deeper lying, decomposing layers. Partially decomposed plant matter is called peat. In some parts of the world, people derive the majority of their fuel from peat bogs. Sphagnum-dominated peatlands cover an area approximately half as large as the United States, more than 1% of Earth's surface. Long ago, continued compression of peat composed primarily of other non-sea plants gave rise to coal. With their simple system of internal transport, the mosses are, in a sense, vascular plants. However, they are not tracheophytes because they lack true xylem and phloem. Introducing the tracheophytes. Although they are an extraordinarily large and diverse group, the tracheophytes can be said to have been launched by a single evolutionary event. Sometime during the Paleozoic era, probably well before the Silurian period, 440 Maya, the sporophyte generation of a now long extinct plant produced a new cell type, the trachid, figure 8. The tracheids is the principal water-conducting element of the xylem in all tracheophytes except the angiosperms, and even in the angiosperms, tracheids persist alongside a more specialized and efficient system of vessels and fibers derived from them. The evolution of a tissue composed of tracheids had two important consequences. First, it provided a pathway for long-distance transport of water and mineral nutrients from a source of supply to regions of need. Second, its stiff cell walls provided something almost completely lacking, and unnecessary, in the largely aquatic green algae, rigid structural support. Support is important in a terrestrial environment because plants tend to grow upward as they compete for sunlight to power photosynthesis. Thus, the tracheids set the stage for the complete and permanent invasion of land by plants. The tracheophytes feature another evolutionary novelty, a branching, independent sporophyte. A branching sporophyte can produce more spores than an unbranched body, and it can develop in complex ways. The sporophyte of a tracheophyte is nutritionally independent of the gametophyte at maturity. Among the tracheophytes, the sporophyte is a large and obvious plant that one normally notices in nature. This pattern is in contrast to the sporophyte of non-tracheophytes such as mosses, which is attached to, dependent on, and usually much smaller than the gametophyte. The present-day evolutionary descendants of the early tracheophytes belong to 7 distinct phyla, see figure 8. The tracheophytes have two types of life cycles, one that involves seeds and another that does not. The non-seed tracheophytes, the two basal phyla, include the club mosses and the ferns and their relatives, horsetails and whisk ferns. Tracheophytes have been evolving for almost half a billion years. The evolution of an effective cuticle and of protective layers for the Gametangia, archegonia, and antheridia helped make the first tracheophytes successful, as did the initial absence of herbivores, plant-eating animals on land. By the late Silurian period, tracheophytes were being preserved as fossils that we can study today. Two groups of non-seed tracheophytes that still exist made their first appearances during the Devonian period, 409 to 354 Maya, the lycopods, club mosses, and the pteridophytes, including horsetails and ferns. Their proliferation made the terrestrial environment more hospitable to animals. Amphibians and insects arrived soon after the plants became established. Trees of various kinds appeared in the Devonian period and dominated the landscape of the Carboniferous. Mighty forests of lycopods up to 40 meters tall, horsetails, and tree ferns flourished in the tropical swamps of what would become North America and Europe. The remnants of those forests are with us today as huge deposits of coal. In the subsequent Permian period, the continents came together to form a single gigantic landmass, called Pangaea. The continental interior became warmer and drier, but late in the period glaciation was extensive. The 200 million year reign of the lycopod fern forests came to an end as they were replaced by forests of sea plants, gymnosperms, which dominated until other sea plants, angiosperms, became dominant less than 80 million years ago. The earliest tracheophytes lacked roots and leaves. The earliest known tracheophytes belonged to the now extinct phylum Rhiniophyta. The Rhiniophytes were among the only tracheophytes in the Silurian period. The landscape at that time probably consisted of bare ground, with stands of rhiniophytes in low-lying moist areas. Early versions of the structural features of all the other tracheophyte phyla appeared in the rhiniophytes of that time. These shared features strengthened the case for the origin of all tracheophytes from a common non-tracheophyte ancestor. In 1917, the British paleobotanist Robert Kitson and William H. Lang reported the finding of well-preserved fossils of tracheophytes embedded in Devonian rocks near Rhynie, Scotland. The preservation of these plants was remarkable, considering that the rocks were more than 395 million years old. These fossil plants had a simple vascular system of phloem and xylem. Some of the plants had flattened scales on the stems, which lacked vascular tissue and thus were not comparable to the true leaves of any other tracheophytes. These plants also lacked roots. They were apparently anchored in the soil by horizontal portions of stem, called rhizomes, that bore water-absorbing rhizoids. These rhizomes also bore aerial branches, and sporangia, homologous with the non-tracheophyte capsule, were found at the tips of these branches. Their branching pattern was dichotomous, that is, the shoot apex divided to produce two equivalent new branches, each pair diverging at approximately the same angle from the original stem, figure 9. Scattered fragments of such plants had been found earlier, but never in such profusion or so well preserved as those discovered by Kitson and Lang. The presence of xylem indicated that these plants were tracheophytes, but were they sporophytes or gametophytes? Close inspection of thin sections of fossil sporangia revealed that the spores were in groups of 4. In almost all living non-seed tracheophytes, with no evidence to the contrary from fossil forms, the 4 products of meiosis and cytokinesis remain attached to one another during their development into spores. The spores separate only when they are mature, and even after separation their walls reveal the exact geometry of how they were attached. Therefore, a group of 4 closely packed spores is found only immediately after meiosis, and a plant that produces such a group must be a diploid sporophyte, and so the Rhynie fossils must have been sporophytes. Gametophytes of the Rhiniophyta were also found. They, too, were branched, and depressions at the apices of the branches contained Archegonia and Antheridia. Although they were apparently ancestral to the other tracheophyte phyla, the Rhiniophytes themselves are long gone. None of their fossils appear anywhere after the Devonian period. Early tracheophytes added new features. A new phylum of tracheophytes, the Lycophyta club mosses, also appeared in the Silurian period. Another, the Pteridophyta ferns and fern allies, appeared during the Devonian period. These two groups arose from Rhiniophyte-like ancestors. These new groups featured specializations not found in the Rhiniophytes, including one or more of the following: true roots, true leaves, and a differentiation between two types of spores. The origin of roots. The Rhiniophytes had only rhizoids arising from a rhizome with which to gather water and minerals. How, then, did subsequent groups of tracheophytes come to have the complex roots we see today? It is probable that roots had their evolutionary origins as a branch, either of a rhizome or of the above ground portion of a stem. That branch presumably penetrated the soil and branched further. The underground portion could anchor the plant firmly, and even in this primitive condition it could absorb water and minerals. The discovery of fossil plants from the Devonian period, all having horizontal stems, rhizomes with both underground and aerial branches, supported this hypothesis. Underground and above ground branches, growing in sharply different environments, were subjected to very different selection pressures during the succeeding millions of years. Thus the two parts of the plant axis, the above ground shoot system and the underground root system, diverged in structure and evolved distinct internal and external anatomies. In spite of these differences, scientists believe that the root and shoot systems of tracheophytes are homologous, that they were once part of the same organ. The origin of true leaves. Thus far we have used the term leaf rather loosely. We spoke of leafy mosses and commented on the absence of true leaves in Rhiniophytes. In the strictest sense, a leaf is a flattened photosynthetic structure emerging laterally from a main axis or stem and possessing true vascular tissue. Using this precise definition as we take a closer look at true leaves in the tracheophytes, we see that there are two different types of leaves, very likely of different evolutionary origins. The first leaf type, the microphyll, is usually small and only rarely has more than a single vascular strand, at least in plants alive today. Plants in the phylum Lycophyta club mosses, of which only a few genera survive, have such simple leaves. The evolutionary origin of microphylls is thought by some biologists to be sterile sporangia, figure 10a. The principal characteristic of this type of leaf is that its vascular strand departs from the vascular system of the stem in such a way that the structure of the stem's vascular system is scarcely disturbed. This was true even in the fossil lycopod trees of the Carboniferous period, many of which had leaves many centimeters long. The other leaf type is found in ferns and seed plants. This larger, more complex leaf is called a megaphyll. The megaphyll is thought to have arisen from the flattening of a dichotomously branching stem system and the development of overtopping, a pattern in which one branch differentiates from and grows beyond the others. This change was followed by the development of photosynthetic tissue between the members of overtopped groups of branches, figure 10b. Megaphylls may have evolved more than once, in different phyla of tracheophytes showing overtopping of branches. Homospory and heterospory. In the most ancient of the present-day tracheophytes, both the gametophyte and the sporophyte are independent and usually photosynthetic. Spores produced by the sporophytes are of a single type, and they develop into a single type of gametophyte that bears both female and male reproductive organs. The female organ is a multicellular archegonium, typically containing a single egg. The male organ is an antheridium, containing many sperm. Such plants, which bear a single type of spore, are said to be homosporous, figure 11a. A different system, with two distinct types of spores, evolved somewhat later. Plants of this type are said to be heterosporous, figure 11b. One type of spore, the megaspore, develops into a larger, specifically female gametophyte, a megagametophite that produces only eggs. The other type, the microspore, develops into a smaller, male gametophyte, a microgametophite that produces only sperm. The sporophyte produces megaspores in small numbers in megasporangia on the sporophyte, and microspores in large numbers in microsporangia. The most ancient tracheophytes were all homosporous, but heterospory evidently evolved independently several times in the early descendants of the Rhiniophytes. The fact that heterospory evolved repeatedly suggests that it affords selective advantages. Subsequent evolution in the plant kingdom featured ever greater specialization of the heterosporous condition. Some tracheophyte clades arose and became extinct in the course of evolution. The earliest clades to arise and survive to this day belong to the non-seed tracheophytes. The surviving non-seed tracheophytes. The non-seed tracheophytes have a large, independent sporophyte and a small gametophyte that is independent of the sporophyte. The gametophytes of the surviving non-seed tracheophytes are rarely more than 1 or 2 centimeters long and are short-lived, whereas their sporophytes are often highly visible. The sporophyte of a tree fern, for example, may be 15 or 20 meters tall and may live for many years. The most prominent resting stage in the life cycle of a non-seed tracheophyte is a single-celled spore. This feature makes their life cycle similar to those of the fungi, the green algae, and the non-tracheophytes, but not, as we will see in the next chapter, to that of the sea plants. Non-seed tracheophytes must have an aqueous environment for at least one stage of their life cycle because fertilization is accomplished by a mobile, flagellated sperm. The ferns are the most abundant and diverse group of non-seed tracheophytes today, but the club mosses and horsetails were once dominant elements of Earth's vegetation. A fourth group, the whisk ferns, contains only 2 genera. In this section we look at the characteristics of these 4 groups and at some of the evolutionary advances that appeared in them. The club mosses are sister to the other tracheophytes. The club mosses and their relatives together called lycopods. Phylum Lycophyta diverged earlier than all other living tracheophytes. That is, the remaining tracheophytes share an ancestor that was not ancestral to the Lycophyta. There are relatively few surviving species of club mosses. The lycopods have roots that branch dichotomously. The arrangement of vascular tissue in their stems is simpler than in the other tracheophytes. They bear only microphylls, and these simple leaves are arranged spirally on the stem. Growth in club mosses comes entirely from apical cell division, and branching is dichotomous, by a division of the apical cluster of dividing cells. The sporangia in many club mosses are contained within cone-like structures called strobili, singular, strobilus, figure 12. Astrobilus is a cluster of spore-bearing leaves inserted on an axis tucked into the upper angle between a specialized leaf and the stem. Such an angle is called an axil. Other club mosses lack strobili and bear the sporangia in the axil between a photosynthetic leaf and the stem. This placement contrasts with the apical sporangia of the Rhiniophytes. There are both homosporous species and heterosporous species of club mosses. Although only a minor element of present-day vegetation, the Lycophyta are one of 2 phyla that appear to have been the dominant vegetation during the Carboniferous period. One type of coal, camel coal, is formed almost entirely from fossilized spores of the tree Lycopod lepidodendron, which gives us an idea of the abundance of this genus in the forests of that time. The other major elements of Carboniferous vegetation were horsetails and ferns. Horsetails, whisk ferns, and ferns constitute a clade. Once treated as distinct phyla, the horsetails, whisk ferns, and ferns form a clade, the phylum. Pteridophyta pteridophytes, or ferns and fern allies. Within that clade, the whisk ferns and the horsetails are both monophyletic, the ferns are not. However, about 97% of all fern species, including those with which you are most likely to be familiar, do belong to a single clade, the Leptosporangia ferns. In the pteridophytes, and in all sea plants, there is differentiation overtopping between the main axis and side branches. Horsetails grow at the bases of stem segments. Like the club mosses, the horsetails are represented by only a few present-day species. All are in a single genus, Equisetum. These plants are sometimes called scouring rushes, because silica deposits found in their cell walls made them useful for cleaning. They have true roots that branch irregularly. Their sporangia curve back toward the stem on the ends of short stalks called sporangiophores, figure 13a. Horsetails have a large sporophyte and a small gametophyte, both independent. The small leaves of horsetails are reduced megaphylls and form in distinct whorls, circles around the stem, figure 13b. Growth in horsetails originates to a large extent from discs of dividing cells just above each whorl of leaves, so each segment of the stem grows from its base. Such basal growth is uncommon in plants, although it is found in the grasses, a major group of flowering plants. Present-day whisk ferns resemble the most ancient tracheophytes. There once was some disagreement about whether Rhiniophytes are entirely extinct. The confusion arose because of the existence today of 2 genera of rootless, spore-bearing plants, Psilotum and Timesicaris, collectively called the whisk ferns. Psilodon has only minute scales instead of true leaves, but plants of the genus Temesiteris have flattened photosynthetic organs, reduced megaphiles, with well-developed vascular tissue. Are these two genera the living relics of the rhiniophytes, or do they have more recent origins? Psilodon and Temesiteris once were thought to be evolutionarily ancient descendants of anatomically simple ancestors. That hypothesis was weakened by an enormous hole in the geological record between the rhiniophytes, which apparently became extinct more than 300 million years ago, and Psilodon and Temesiteris, which are modern plants. DNA sequence data finally settled the question in favor of a more modern origin of the whisk ferns from fern-like ancestors. These two genera are clade of highly specialized plants that evolved fairly recently from anatomically more complex ancestors by loss of complex leaves and true roots. Whisk fern dainophytes live below the surface of the ground and lack chlorophyll. They depend upon fungal partners for their nutrition. Ferns evolve large, complex leaves. The sporophytes of the ferns, like those of the sea plants, have true roots, stems, and leaves. Their leaves are typically large and have branching vascular strands. Some species have small leaves as a result of evolutionary reduction, but even these small leaves have more than one vascular strand and are thus megaphiles. The ferns constitute a group that first appeared during the Devonian period and today consists of about 12,000 species. The ferns are not a monophyletic group, although, as already mentioned, 97% of the species, the Leptosporangia ferns, do constitute a monophyletic group. The Leptosporangia ferns differ from the other ferns in having sporangia with walls only one cell thick, borne on a stalk. Ferns are characterized by fronds, large leaves with complex vasculature. During its development, the fern frond unfurls from a tightly coiled fiddlehead. Some fern leaves become climbing organs and may grow to be as much as 30 meters long. Because they require water for the transport of the male gametes to the female gametes, most ferns inhabit shaded, moist woodlands and swamps. Tree ferns can reach heights of 20 meters. Tree ferns are not as rigid as woody plants, and they have poorly developed root systems. Thus, they do not grow in sites exposed directly to strong winds, but rather in ravines or beneath trees and forests. The sporangia of ferns are found on the undersurfaces of the fronds, sometimes covering the whole undersurface and sometimes only at the edges. In most species, the sporangia are found in clusters called sporoids singular, Soros figure 14. The sporophyte generation dominates the fern life cycle. Inside the sporangia, ferns form mother cells undergo meiosis to form haploid spores. Once shed, the spores travel great distances and eventually germinate to form independent dainophytes. Old world climbing fern, Lygodium microphyllum, is currently spreading disastrously through the Florida Everglades, choking off the growth of other plants. This rapid spread is testimony to the effectiveness of windborne spores. Fern dainophytes have the potential to produce both Antheridia and Archegonia, although not necessarily at the same time or on the same dainophyte. Sperms swim through water to Archegonia, often to those on other dainophytes, where they unite with an egg. The resulting zygote develops into a new sporophyte embryo. The young sporophyte sprouts a root and can thus grow independently of the gametophyte. In the alternating generations of the fern, the gametophyte is small, delicate, and short-lived, but the sporophyte can be very large and can sometimes survive for hundreds of years. Figure 15. Most ferns are homosperous. However, two groups of aquatic ferns, the Marcelisae and Salviniaceae, are derived from a common ancestor that evolved heterospory. The megaspores and microspores of these plants, which germinate to produce female and male dainophytes, respectively, are produced in different sporangia, megasporangia and microsporangia, and the microspores are always much smaller and greater in number than the megaspores. A few genera of ferns produce a tuberous, fleshy gametophyte instead of the characteristic flattened, photosynthetic structure produced by most ferns. Like the dainophytes of whisk ferns, these tuberous dainophytes depend on a mutualistic fungus for nutrition. In some genera, even the sporophyte embryo must become associated with the fungus before extensive development can proceed. The seed plants. The most recent group to appear in the evolution of the tracheophytes is the seed plants. The earliest fossil evidence of seed plants is found in Devonian rocks. The earliest seed plants combine characteristics of rhiniophytes and heterosporous ferns, but they had trachees of the type found in modern seed plants. They also differed from the plants around them by having extensively thickened woody stems, which resulted from the proliferation of xylem. This type of growth in the diameter of stems and roots is called secondary growth. By the Carboniferous period, new lines of seed plants had evolved, including various seed ferns, which possessed fern-like foliage but had seeds attached to their leaves. Two clades of early seed plants are known only as fossils. These clades are basal to the surviving seed plants, which fall into two groups: the gymnosperms such as pines and cycads, and the angiosperms flowering plants. There are 4 living phyla of gymnosperms and 1 of angiosperms. Figure 16. The phylogenetic relationships among these 5 clades have not yet been resolved. All living gymnosperms and many angiosperms show secondary growth. The life cycles of all seed plants share major features, as we are about to see. Seed plants are heterosporous and have tiny dainophytes. In seed plants, the gametophyte generation is reduced even further than it is in the ferns. Figure 17. The haploid gametophyte develops partly or entirely while attached to a nutritionally dependent on a diploid sporophyte. Among the seed plants, only the earliest types of gymnosperms and a few survivors had swimming sperm. All other seed plants have evolved other means of bringing eggs and sperm together. The culmination of this striking evolutionary trend was independence from the liquid water that earlier plants needed for sexual reproduction. Seed plants are heterosporous. They form separate megasporangia and microsporangia on structures that are grouped on short axes, such as the cones and strobili of conifers and the flowers of angiosperms. As in other plants, the spores of seed plants are produced by meiosis within the sporangia, but in seed plants, the megaspores are not shed. Instead, they develop into female dainophytes within the megasporangia. These megadainophytes are dependent on the sporophyte for food and water. In most seed plant species, only one of the meiotic products in a megasporangium survives. The surviving haploid nucleus divides mesotically, and the resulting cells divide again to produce a multicellular female gametophyte. This megadainophyte is retained within the megasporangium, where it matures. The megadainophyte, in turn, houses the early development of the next sporophyte generation following fertilization of the egg. The megasporangium is surrounded by sterile sporophytic structures that form a protective integument. Within the microsporangium, the meiotic products are microspores, which divide mesotically within the spore wall one or a few times to form a male gametophyte called a pollen grain. Pollen grains are released from the microsporangium to be distributed by wind, an insect, a bird, or a plant breeder. Figure 18. A pollen grain that reaches the appropriate surface of a sporophyte of the same species develops further. It produces a slender pollen tube that elongates and digests its way through the sporophytic tissue toward the female gametophyte. When the tip of the pollen tube reaches the female gametophyte, sperm are released from the tube, and fertilization occurs. The resulting diploid zygote divides repeatedly, forming a young sporophyte that develops to an embryonic stage at which growth is temporarily suspended, often referred to as a dormant stage. The end product at this stage is a multicellular seed. The seed is a complex package. A seed may contain tissues from 3 generations. The seed coat develops from tissues of the diploid sporophyte parent, the integument. Within the megasporangium is a haploid female gametophytic tissue from the next generation, which contains a supply of nutrients for the developing embryo. This tissue is fairly extensive in most gymnosperm seeds. In angiosperm seeds, its place is taken by a tissue called endosperm, which we will describe below. In the center of the seed is the 3rd generation, the embryo of the new diploid sporophyte. The seed of a gymnosperm or an angiosperm is a well-protected resting stage. The seeds of some species may remain viable, capable of growth and development for many years, germinating when conditions are favorable for the growth of the sporophyte. In contrast, the embryos of non-seed plants develop directly into sporophytes, which either survive or die, depending on environmental conditions. There is no dormant stage in the life cycle. During the dormant stage, the seed coat protects the embryo from excessive drying and may also protect it against potential predators that would otherwise eat the embryo and its nutrient reserves. Many seeds have structural adaptations that promote their dispersal by winter, more often by animals. When the young sporophyte resumes growth, it draws on the food reserves in the seed. The possession of seeds is a major reason for the enormous evolutionary success of the seed plants, which are the dominant life forms of Earth's modern terrestrial flora in most areas. The gymnosperms, naked seeds. The extant gymnosperms are a clade of seed plants that do not form flowers. Although there are probably fewer than 750 species of living gymnosperms, these plants are second only to the angiosperms in their dominance of the terrestrial environment. There are 4 clades of living gymnosperms today. The cycads phylum Cycadophyta are palm-like plants of the tropics and subtropics, growing as tall as 20 meters. Of the present-day gymnosperms, the cycads are probably closest to the earliest seed plants. Ginkgos phylum Ginkgophyta, which were common during the Mesozoic era, are represented today by a single genus and species, Ginkgo biloba, the maidenhair tree. There are both male microsporangia and female megasporangia, maidenhair trees. The difference is determined by X and Y sex chromosomes, as in humans, few other plants have sex chromosomes. The phylum Nymphaea consists of 3 very different genera that share certain characteristics with the angiosperms. One of the Nymphaeae is Welwitschia, a long-lived desert plant with just 2 strap-like leaves that sprawl on the sand and can grow as long as 3 meters. By far the most abundant of the gymnosperms are the conifers, phylum Pinophyta, cone-bearing plants such as pines and redwoods. All living gymnosperms except the Nymphaea have only trachees as water-conducting and support cells in their xylem. They lack the more specialized vessels and fibers found alongside trachees in the angiosperms. Although this difference may make the gymnosperm water transport and support system seem less efficient than that of the angiosperms, it serves some of the largest trees known. The coast redwoods of California are the tallest gymnosperms. The largest are well over 100 meters tall. Secondary xylem, wood, produced by gymnosperms, is the principal resource of the timber industry. During the Permian period, the conifers and cycads flourished. Gymnosperm forests changed over time as the gymnosperm groups evolved. Gymnosperms dominated the Mesozoic era, during which the continents drifted apart and dinosaurs strode the earth. They were the principal trees in all forests until less than 100 million years ago, and they still dominate many present-day forests. Let's look at the most abundant gymnosperms, the conifers, in more detail. Conifers have cones but no motile cells. The Great Douglas fir and cedar forests of the northwestern United States and the massive boreal forests of pine, fir, and spruce found in northern regions of Eurasia and North America, as well as on the upper slopes of mountain ranges everywhere, rank among the great vegetation formations of the world. All these trees belong to one phylum of gymnosperms, Pinophyta. The conifers are cone bearers. A cone is a short axis, a modified stem bearing a tight cluster of scales, which are reduced branches specialized for reproduction. Figure 19a. A strobilus is a cone-like cluster of scales that are modified leaves inserted on an axis. Figure 19b. Megaspores are produced in seed cones, and microspores are produced in pollen strobili. Seed cones are much larger than pollen strobili. We will use the life cycle of a pine to illustrate reproduction in gymnosperms. Figure 20. The production of male dainophytes in the form of pollen grains frees the plant completely from its dependence on liquid water for fertilization. Instead of water, wind assists conifer pollen grains in the first stage of travel from the strobilus to the female gametophyte inside the seed cone. The pollen tube provides the sperm with the means for the last stage of travel by elongating and digesting its way through maternal sporolytic tissue. When it reaches the female gametophyte, it releases two sperm, one of which degenerates after the other unites with an egg. The megasporangium, in which the female gametophyte will form, is enclosed in a layer of sporophytic tissue, the integument, that will eventually develop into the seed coat. The integument, the megasporangium inside it, and the tissue attaching it to the maternal sporophyte constitute the ovule. The pollen grain enters through a small opening in the integument at the tip of the ovule, the micropyle. Gymnosperms derive their name, which means not heat-seeded, from the fact that their ovules and seeds are not protected by ovary or fruit tissue. Most conifer ovules which, upon fertilization, develop into seeds, are born exposed on the upper surfaces of the modified branches that form the scales of the cone. Each cone scale lies in the angle between the modified leaf and the axis. The only protection of the ovules comes from the scales, which are tightly pressed against each other within the cone. As we have seen, some pines, such as the lodgepole pine, have such tightly closed seed cones that only fire suffices to split them open and release the seeds. About half of the conifer species have soft, fleshy fruit-like tissues associated with their seeds. Examples are the fleshy cones or berries of juniper and yew. Animals may eat these tissues and then disperse the seeds in their feces, often carrying them considerable distances from the parent plant. These tissues, however, are not true fruits, which are characteristic of the plant phylum that is dominant today, the angiosperms. The angiosperms, flowering plants. The phylum Angiospermae consists of the flowering plants, also commonly known as the angiosperms. This highly diverse phylum includes more than 257,000 species. The oldest evidence of angiosperms dates back to the early Cretaceous period, about 140 million years ago. The angiosperms radiated explosively and, over a period of only about 60 million years, became the dominant plant life of the planet. In later chapters, when we mention plants, we are generally referring to the angiosperms. The female gametophyte of the angiosperms, consisting of just 7 cells, is even more reduced than that of the gymnosperms. Thus, the angiosperms represent the current extreme of an evolutionary trend that runs throughout the trichiophytes. The sporophyte generation becomes larger and more independent of the gametophyte, while the gametophyte generation becomes smaller and more dependent on the sporophyte. A number of synapomorphies, shared derived traits, characterize the angiosperms. They have double fertilization. They produce a triploid nutritive tissue called the endosperm. Their ovules and seeds are enclosed in a carpel. They have flowers. They produce fruit. Their xylem contains vessel elements and fibers. Their phloem contains companion cells. Double fertilization was long considered the single most reliable distinguishing characteristic of the angiosperms. Two male gametes, contained within a single microgametophyte pollen grain, participate in fertilization events within the megagametophyte of an angiosperm. One sperm combines with the egg to produce a diploid zygote, the first cell of the sporophyte generation. In most angiosperms, the other sperm nucleus combines with two other haploid nuclei of the female gametophyte to form a triploid 3-end nucleus. This nucleus, in turn, divides to form a triploid tissue, the endosperm, that nourishes the embryonic sporophyte during its early development. Double fertilization occurs in nearly all present-day angiosperms. We are not sure when and how it evolved because there is no known fossil evidence on this point. It may have first resulted in two embryos, as it does in the three existing genera of Nidophyta, Ephedra, Nenum, and Velbeckia. Both of the fertilizations in Nidophytes produce diploid products. The name angiosperm, enclosed seed, is drawn from another distinctive character of these plants. The ovules and seeds are enclosed in a modified leaf called a carpel. Besides protecting the ovules and seeds, the carpel often interacts with incoming pollen to prevent self-pollination, thus favoring cross-pollination and increasing genetic diversity. Of course, the most evident diagnostic feature of angiosperms is that they have flowers. Production of a fruit is another of their unique characteristics. Most angiosperms are also distinguished by the possession of specialized water-transporting cells called vessel elements in their xylem, but these cells are also found in anatomically different form in Nidophytes and a few ferns. A second distinctive cell type in angiosperm xylem is the fiber, which plays an important role in supporting the plant body. Angiosperm phloem possesses another unique cell type called the companion cell. Like the gymnosperms, woody angiosperms show secondary growth, producing secondary xylem and secondary phloem and growing in diameter. In the following sections, we will examine the structure and function of flowers, evolutionary trends in flower structure, the functions of pollen and fruits, the angiosperm life cycle, the two major groups of angiosperms, and the origin and evolution of flowering plants. The sexual structures of angiosperms or flowers. If you examine any familiar flower, you will notice that the outer parts look somewhat like leaves. In fact, all the parts of a flower are modified leaves. A generalized flower, for which there is no exact counterpart in nature, is diagrammed below for the purpose of identifying its parts. The structures bearing microsporangia are called stamens. Each stamen is composed of a filament bearing an anther that contains pollen-producing microsporangia. The structures bearing megasporangia are the carpels. A structure composed of one carpel or two or more fused carpels is called a pistil. The swollen base of the pistil, containing one or more ovules, each containing a megasporangium surrounded by its protective integument, is called the ovary. The apical stalk of the pistil is the style, and the terminal surface that receives pollen grains is the stigma. In addition, a flower often has several specialized sterile, non-spore ringed leaves. The inner ones are called petals collectively, the corolla, and the outer ones sepals collectively, the calyx. The corolla and calyx, which can be quite showy, often play roles in attracting animal pollinators to the flower. The calyx more commonly protects the immature flower in bud. From base to apex, the sepals, petals, stamens, and carpels, which are referred to as the floral organs, are usually positioned in circular arrangements or whorls and attached to a central stalk called the receptacle. The generalized flower shown in figure has both megasporangia and microsporangia. Such flowers are referred to as perfect. Many angiosperms produce two types of flowers, one with only megasporangia and the other with only microsporangia. Consequently, either the stamens or the carpels are non-functional or absent in a given flower, and the flower is referred to as imperfect. Species such as corner birch, in which both megasporangia and microsporangia flowers occur on the same plant, are said to be monoecious, meaning one house, but it must be added, one house with separate rooms. Complete separation is the rule in some other angiosperm species, such as willows and date palms. In these species, a given plant produces either flowers with stamens or flowers with pistils, but never both. Such species are said to be dioecious, two-housed. Flowers come in an astonishing variety of forms, as you will realize if you think of some of the flowers you recognize. The generalized flower shown in figure has distinct petals and sepals arranged in distinct whorls. In nature, however, petals and sepals sometimes are indistinguishable. Such appendages are called tepals. In other flowers, petals, sepals, or tepals are completely absent. Flowers may be single, or they may be grouped together to form an inflorescence. Different families of flowering plants have their own characteristic types of inflorescences, such as the compound umbels of the carrot family, the heads of the aster family, and the spikes of many grasses. Flower structure has evolved over time. The flowers of the most basal lineages of angiosperms have a large and variable number of tepals, or sepals and petals, carpels, and stamens. Evolutionary change within the angiosperms has included some striking modifications of this early condition, reductions in the number of each type of floral organ to a fixed number, differentiation of petals from sepals, and changes in symmetry from radial, as in a linear magnolia, to bilateral, as in a sweet pea orchid, often accompanied by an extensive fusion of parts. According to one theory, the first carpels to evolve were modified leaves, folded but incompletely closed, and thus differing from the scales of the gymnosperms. In the groups of angiosperms that evolved later, the carpels fused and became progressively more buried in receptacle tissue, figure 21a. In the flowers of the most recent groups, the other flower parts are attached to the very top of the ovary, rather than at the bottom. The stamens of the most ancient flowers may have appeared leaf-like, figure 21b, little resembling those of the generalized flower in figure. Why do so many flowers have pistils with long styles and anthers with long filaments? Natural selection has favored length in both of these structures, probably because length increases the likelihood of successful pollination. Long filaments may bring the anthers into contact with insect bodies, or they may place the anthers in a better position to catch the wind. Similar arguments apply to long styles. A long style may serve another purpose as well. If several pollen grains land on one stigma, a pollen tube will start growing from each grain down the style toward the ovary. If there are more pollen grains than ovules, there is a race to fertilize the ovules. The race down the style can be viewed as mate selection by the plant bearing the style. Angiosperms have co-evolved with animals. Pollen has played another crucial role in the evolution of the angiosperms. Whereas many gymnosperms are wind-pollinated, most angiosperms are animal-pollinated. Animals visit flowers to obtain nectar or pollen, and in the process often carry pollen from one flower to another, or from one plant to another. Thus, in its quest for food, the animal contributes to the genetic diversity of the plant population. Insects, especially bees, are among the most important pollinators. Birds and some species of bats also play major roles as pollinators. For more than 130 million years, angiosperms and animal pollinators have co-evolved in the terrestrial environment. The animals have affected the evolution of the plants, and the plants have affected the evolution of the animals. Flower structure has become incredibly diverse under these selection pressures. Some of the products of coevolution are highly specific. For example, some species are pollinated by only one species of moth. Pollination by just one or a few animal species provides a plant species with a reliable mechanism for transferring pollen from one of its members to another. Most plant-pollinator interactions are much less specific. That is, many different animal species pollinate the same plant species, and the same animal species pollinate many different plant species. However, even these less specific interactions have developed some specialization. Bird-pollinated flowers are often red and odorless. Many insect-pollinated flowers have characteristic odors, and bee-pollinated flowers may have conspicuous markings or nectar guides that are evident only in the ultraviolet region of the spectrum, where bees have better vision than in the red region. The angiosperm life cycle features double fertilization. The life cycle of the angiosperms is summarized in Figure 22. Like all sea plants, angiosperms are heterosporous. The ovules are contained within carpels, rather than being exposed on the surfaces of scales, as in most gymnosperms. The male gametophytes, as in the gymnosperms, are pollen grains. The ovule develops into a seed containing the products of the double fertilization that characterizes angiosperms, a diploid zygote and a triploid endosperm. The endosperm serves as storage tissue for starch or lipids, proteins, and other substances that will be needed by the developing embryo. The zygote develops into an embryo, consisting of an embryonic axis and one or two cotyledons, or seed leaves. The cotyledons have different fates in different plants. In many, they serve as absorptive organs that take up and digest the endosperm. In others, they enlarge and become photosynthetic when the seed germinates. Often they play both roles. Angiosperms produce fruits. The ovary of a flowering plant, together with the seeds it contains, develops into a fruit after fertilization. A fruit may consist only of the mature ovary and its seeds, or it may include other parts of the flower structures associated with it. A simple fruit, such as a cherry, is one that develops from a single carpel or several united carpels. A raspberry is an example of an aggregate fruit, one that develops from several separate carpels of a single flower. Pineapples and figs are examples of multiple fruits, formed from a cluster of flowers and inflorescence. Fruits derived from parts in addition to the carpel and seeds are called accessory fruits. Examples are apples, pears, and strawberries. There are several clades of angiosperms. The better understood relationships among the angiosperm clades are shown in Figure 23. Two large clades include the great majority of angiosperm species, the monocots and the eudicots. The monocots are so called because they have a single embryonic cotyledon. The eudicots have two. Some familiar angiosperms belong to clades other than the monocots and eudicots. These clades include the water lilies, star anise and its relatives, and the magnolia complex. The magnolias are less numerous than the monocots and eudicots, but they include many familiar and often useful plants such as magnolias, avocados, cinnamon, and pepper. The monocots include grasses, cattails, lilies, orchids, and palms. The eudicots include the vast majority of familiar seed plants, including most herbs, vines, trees, and shrubs. Among them are such diverse plants as oaks, willows, violets, snapdragons, and sunflowers. Determining the oldest angiosperm clade. Which angiosperms were the earliest flowering plants was long a matter of great controversy. Two leading candidates were the magnolia family and another family, the Chloranthaceae, whose flowers are much simpler than those of the magnolias. At the close of the 20th century, however, an impressive convergence of evidence led to the conclusion that the most basal living angiosperm belongs to neither of those families, but rather to a clade that today consists of a single species of the genus Ambrella. This woody shrub, with cream-colored flowers, lives only on New Caledonia, an island in the South Pacific. Its 5 to 8 carpels are in a single whorl, and it has 30 to 100 stamens. The xylem of Ambrella lacks vessel elements, which appeared later in angiosperm evolution. The characteristics of Ambrella give us a good sense of what the first angiosperms might have been like. But are there extinct angiosperms that may represent still more ancient clades? In 2002, Chinese and American botanists examined fossils of two species of the 125-million-year-old aquatic genus Archaeophractus. Their studies established an extinct family, Archaeophractaceae, that is posited to be the sister taxon of all other angiosperms. The flower of these plants had its ovules enclosed in carpels, as in all angiosperms. The flower had neither petals nor sepals, however, and its carpels and stamens were arranged spirally around elongated shoots. This arrangement of carpels and stamens is seen today in the magnolias. The origin of the angiosperms remains a mystery. We have learned a lot about evolution within the angiosperm clade, but how did the angiosperms first arise? Are the angiosperms sister to any single gymnosperm phylum? A few years ago, it seemed that we were on the verge of answering these questions, but the puzzle remains as vexing today as it ever was. Why should this be? Different phylogenetic methods, applied by different investigators, have produced apparently contradictory results. It might seem a simple matter to rectify this situation, but several questions complicate such efforts. What morphological characters should be selected is important, or should they all be treated? It might seem a simple matter to rectify this situation, but several questions complicate such efforts. What morphological characters should be selected is important, or should they all be treated as equally important? What algorithms should be applied to computerized analysis of data? Are all molecular differences and similarities significant, or are some of them incidental? Which fossils should be chosen for comparisons? What is the likelihood that we can find evidence of double fertilization in ancient fossils? Furthermore, it is possible that the angiosperms have no close relatives at all among living sea plants. We are left with our original question: where did the first angiosperm come from? Current progress in methodology gives us reason to hope that our understanding of sea plant evolution will be much improved before the present decade ends. Animals, descendants of a common ancestor. Biologists have long debated whether animals